Ben O'Shaughnessy from Columbia University. Ben is, I would say, a physicist. His character means something. And he has been doing quite interesting work about what concerns vesicular release through the modeling of this snare complex. So, Ben, thank you.
Thank you, David. So, Krasi and David, you're probably fed up with being thanked. So I will thank you both again for organizing this very interesting workshop. And I think, yeah, everybody's learning a great deal. I'm learning with every talk and every question session, and it's been really great. And thank you for inviting me to participate. So, let me just see if I can master the equipment. So we've had there's the synapse, and we've had a number of presynaptic talks, a number of postsynaptic talks, and I guess some people have straddled the two. I can't quite remember now, but this talk is definitely presynaptic, and what I will talk about is what's going on in the presynaptic terminal during evoked release, which may be, of course, in the real world an actual in the brain, in the central nervous system, and elsewhere, an action potential, or it may be evoked in experimental situations by calcium uncaging. So as Mathieu and Fred, they gave very nice introductions to aspects of what this cartoon is all about. So synaptic vesicles get tethered and docked and primed. That's the language. And in this primed state, there is a machinery which docks the vesicle very close to the membrane and is ready to receive calcium when an action potential comes along and activates voltage-gated calcium channels. Calcium enters, and then something happens, something very complicated and something remarkable as well, because this machinery on sub-millisecond timescales does this. It fuses the vesicular membrane and plasma membranes and then releases neurotransmitter. So I'm talking about what's in the red box. I will talk little, what we've been trying to understand does not concern this. The calcium will be a given. Obviously it's very important. And Julia, for example, and Krasi, you discussed in detail the importance of how the calcium gets to have the time profile that it has, but that'll be a given for us. What I'll be talking about is this machinery. Right from here, the instant the calcium comes in, to that moment when release happens. And I won't even talk about this, and this is all very important as well, how the vesicle merges with the plasma membrane, and there's the fate of the vesicle subsequently can be to get recycled, or full fusion can occur, and so on. So this is a complex machinery. It's not just a fusion machinery, because it's a fusion machinery which can sense calcium and then somehow get unclamped, and then fuse the membranes. But it's well established that the core of the fusion machinery, in some sense, are the snare proteins. Mathieu and Fred gave us nice introductions to that. I'll just briefly remind you, for those of you who need reminding, that the vesicle carries V snares, one of these blue guys. T snares reside in the plasma membrane, and they kind of hook up and zip it down. And there's a number of them, and the number is very controversial. How many are needed, if that's the right language, and I will question that language. But how many are there is very controversial. Anyway, there's a bunch of them, and somehow the membranes get pulled together, and they fuse. And even that core aspect, I think it's fair to say, is poorly understood. It's not the main point of this talk, really. This is just part of what I want to talk about. I want to talk about mainly our efforts to model the entire machinery, our first steps in that direction. So there are many other components in this machinery that have been identified. Synaptotagmin is known to be the calcium sensor. We've heard a lot about synaptotagmin already. Munc18 involved in assembly. Munc13 is another one. Complexin is known to be involved. Complexin seems to clamp spontaneous fusion, but to have a promoting role for evoked fusion. So there are many components in this machinery, and this is an exciting stage for this field, because the big question really is, I mean, there's a whole list of questions, but perhaps you could say the biggest question is now, how do these things fit together? How do they work as a machine? And of course, that's interesting for modeling types, because we tend to like to think of collective effects and how machines work, rather than individual proteins. You have to really think about the individual proteins, of course. And various proposals have been made. For example, these are from the Brunner lab. So this is a cartoon. They're all cartoons. They're all, the product of imaginative thinking. Nobody knows no one knows how these pieces are put together. But for example, here's one proposal. There's the so-called snare pin. So that's what the snare complex is called when the blue vamp, the blue V snare, and the red syntaxin and the green SNAP25 helices complex together. Two synaptotagmins are shown here, corresponding to two interfaces that have been determined by this group from X-ray crystallography. The top one involving complexin as well. Fred briefly alluded to that. That's one possibility, that maybe they're sitting in this upright configuration, and then somehow when calcium comes along, something happens to the synaptotagmin and pulls them apart in some way. Another proposal by these guys is there's a side view, here's a top view, that maybe the synaptotagmins laterally bridge the snares. An interesting proposal from the Rothman group is that synaptotagmin, by virtue of its ability to form rings, so the Rothman lab showed that synaptotagmin proteins spontaneously oligomerize into rings. And for that oligomerization to occur, a charged lipid bilayer is needed, a membrane is needed. Actually, they first discovered this with monolayers. And the interesting thing is that when calcium comes along, the ring falls apart. So you could immediately imagine that suggests an interesting mechanism there. Maybe the ring is around to, and this was Jim Rothman's proposal. What the ring does is it acts as a spacer. It clamps fusion, in other words. That's the language. Again, I think Fred used that terminology yesterday. Clamps fusion, there's a brake on the fusion apparatus, and if you were to release that brake, it would fuse. So the idea is that this clamping mechanism is as a spacer. Washer hypothesis is another language that Jim Rothman et al. Used. Then calcium comes along, disassembles the ring, and that allows fusion to take place. So actually, the trajectory of this talk is that I'll describe our efforts generally to make a modeling system which can test different machineries. And we think that's a very important thing for the field. That's badly needed. It's an exciting thing to be able to do if you can do it. It's very ambitious. And I'll describe our efforts to test this hypothesis in its simplest form. But I don't mean to suggest that we're wedded to that hypothesis. You know, we have a very nice collaboration with Jim, and we've been following their thinking and their experiments on this topic. And it's a very exciting proposal, and I think that's the way to think. You know, this is the way the machine works. Let's test it. Let's try it. And that's a kind of a new way of thinking in the field. How do all the components work together? Not that the identification and the characterization of individual components is not continuing and will not continue. Of course it will. But perhaps, you know, the overarching issue now is how do they all fit together? So let's say you did want to embark on this adventurous modeling challenge to actually try to reconstitute, if you like, this is a language we sometimes use, to try to sort of reconstitute theoretically or in silico, if you like, the fusion machinery. So how would one go about that? The first thing you might think is let's just put all of the molecules on a computer, represent them in all atom representations, and that's a very well-developed field, and it makes very important contributions to this and many other fields, of course. So that might be the first sort of thing you'd say. Let's just, you know, put them on a computer and see what happens. There's a problem with doing that, and that's because of the timescales. So we've seen in a number of talks in the last few days, we've been exposed to what those timescales are. But if you look at measurements, say at fast CNS synapses or calyx of Held, these retinal bipolar cells, these guys are, you know, as you know, giant synapses that release many vesicles when an action potential comes along. But if you look at the various measurements, the time scales, well, the smallest time scale on this slide just about is that one, 60 microseconds. There's a millisecond there. There's a millisecond there. This is inverse time. There's 10 milliseconds there. There's even a tenth of a second there. And the same kind of story in these experiments. So these timescales computationally, at least from the point of view of an all-atom representation of the different molecules, are daunting. In fact, they're impossible. Particularly because you can see from some of these cartoons that I've shown you already that many molecules are involved. We're not talking about just simulating one snare. That's hard enough to do for longer than a kind of a microsecond or time scale. All atom-wise, with all atom methods, one's more in the nanoscale, the nanosecond range. Yet another issue is, I wouldn't say this is actually the biggest issue, but it's relevant nevertheless, that release probabilities tend to be rather low. I mean, they're all over the map, but what does that mean? That means if you try to simulate what happens to one vesicle, you've got to do 10 or 30 or 100 simulations to get one event. So that's another factor that's making things harder. As I say, I wouldn't say it's actually the dominant one. So conventional simulation approaches, as I muttered a few seconds ago, all atom, you can typically get nanosecond timescales. More coarse-grained, well-known methods like Martini, so they're somewhat coarse-grained, and you can maybe get up to microsecond timescales. And again, this is with a relatively small number of molecules. So our approach to this problem has been what perhaps I can call radical coarse graining. So we go as far as we can possibly go in our judgment without, you know, throwing the baby out with the bathwater, as they say. So let's take the snares. We coarse grain the snares to the degree that, so here's the real thing, the crystal structure. There's our coarse-grained version. So there are beads representing four residues, which corresponds roughly to one of the 16 layers in the snare complex. So it's radical, but we we preserve as much of the biochemical and biophysical reality as is possible. So for example, the beads, they obviously have the right size, they're carefully, spatially arranged, the coarse graining is very systematic, they have the charge corresponding to the residues that they represent. So the model I'll describe, first let me mention,
Ultimately, you end up with how many beads in the coarse graining of those molecules?
Well, I'd have to do a bit of mental arithmetic, which I'm incapable
it's like
of.
10, 100,000.
Well, let's look at this guy. So there are 16 layers. So maybe you're asking a different question, but let me start by looking at this snare here. So we've got 16 beads times 4, 64, times, let's say, 10 snares. So that's getting up to about 1,000, and then you have all the other molecules. But we don't represent the membranes. That's a daunting task.
And the goal of all of
these... sorry to interrupt you, David, but we do represent the membranes. We don't represent them in a molecular way. Yeah.
And so where you are aiming now is to understand how these snare interact in order to go from the configuration or conformation where they are like this to the configuration where the vesicle will
Well, the story
open.
isn't, but it's much more than the snares. There's a whole machinery. So we'll see pictures, and then perhaps you can come back with questions about that point at that stage, David. Our membranes are, this is a lie, this picture here. This is just a pretty picture in this and this part of the cartoon, because we don't have a bunch of lipids, we just have fixed surfaces. But we do have all of the interactions that are known. Van der Waals, electrostatic, hydration forces are very important, as was briefly discussed the other day. When two hydrated lipid surfaces get very close to one another, they feel a well-characterized hydration force. I mean, well-characterized in these important experiments that years ago Rand and Possegian performed. We used the zippering energy landscape measured by Yongli Zhang with optical tweezers, and sorry, I'm being a little incompetent with this device. So just to give a sense of the degrees of freedom, any amount of zippering can occur, or unzippering. That degree of freedom is allowed to equilibrate. It's constantly changing in general. It's constantly fluctuating. No assumptions about that. And perhaps I should just say at this point that our aim is to have no assumptions in there. Obviously that's impossible, because unless you have the real molecules, you're making assumptions about them. But as much as possible, we try to be agnostic. Just to take, just like the all-atom people try to be agnostic. They don't want to make any, there's no model there, right? Theoretically. They just want to take the crystal structure, which is measured experimentally, and then represent it faithfully on a computer. That's the objective. Of course, there's always got to be some assumptions in doing that, but that's what we're striving for here. So the phenomena and the mechanisms that emerge are not put in any sense by us. They emerge. I think that's an important point to make. And perhaps I'll also say this: that many speakers have apologized for being either experimentalists or not being experimentalists, or not having equations or having lots of equations. So here's my apology: no equations. And I can't believe I'm giving this talk. I never used to do computational analysis at all. It was all analytical. And so two responses to the question I've just put to myself in a way there. I think it's very important for this class of problems to do this level of analysis. And what we've found is that the insight we've got from these types of simulation have actually spurred us into analytical approaches. If you invite me back, maybe next year, or whenever it is, I'll tell you about some of those. But anyway, so of course, it's not just the snares. Far from it. The snares are just one of many components in the machinery. So our goal is to I mean, we've published work with the snares coarse-grained in these kinds of ways. Our goal is to incorporate other components. Beginning with synaptotagmin, it's the first one you would try to incorporate because it's a calcium sensor. And if you don't have that, how can you hope to reproduce these evoked release phenomena? So Aniban Poli and Zach McDarg, postdocs in my group, have, particularly for this aspect, Aniban developed a systematic coarse graining scheme for other components of the machinery. So synaptotagmin, this is the all-atom representation from the crystal structure. This is our coarse-grained version. There is a critical C2A domain and a C2B domain. And there are a bunch of calcium binding sites, and I'll say more about that. So a similar systematic coarse graining scheme. I mentioned that synaptotagmin is known to oligomerize into rings under the right conditions. So we using the our coarse-grained version of the crystal structure for synaptotagmin, and what we know from electron microscopy studies from the Rothman group of these rings, we assemble them into the 20 synaptotagmins into a ring. In 2015, the Brunger group published this very important crystal structure. So crystal structures are very important announcements in this field, because you got all these bits and pieces, and you think, God, how are they put together? What's the anatomy? What's the architecture? So every new announcement is a very exciting event. And of course, you have to take some, I'm no expert on that. I'm on a learning curve, sort of down there somewhere. But it is a crystal, so you've got to be a little suspicious always of possible artifacts and so on. But what they found in this 2015 structure was a binding site between the C2B domain of synaptotagmin and the snare complex, with a particular orientation. So we reproduce that as precisely as we can in this structure, and that produces an orientation of the snares which is close to, I forget the angle. It's not tangential, it's oriented in at some angle, which I can't remember, 40 degrees or something. So it's not way off. It's not very far off tangential, but...
Ben, can you explain again your method of coarse graining? You just replace a piece of a molecule by a ball with the associated charge. Or there is something else?
The size is respected, and the charge is respected, and the location is respected, and that's it.
But about the geometry, the organization,
So the positioning is in very precise correspondence to the crystal structure.
And then it is frozen at a certain configuration?
Well, it depends on the degree of freedom. For example, remember that this can unzipper, and certainly this can unzipper. So it's as dynamic a structure as you want to make it. But admittedly, it is a crude representation. So you can imagine, it depends on the community, but you can imagine the kind of responses that we get from some reviewers, because it's so radically coarse-grained. It's not from the relative to a partial differential equation representation, but relative to an all-atom representation, it is. So. But what we say is that you may say that it's coarse-grained, and that is I think this. How you coarse grain is something that is critical, and it's a developing field in many areas of science. What's the best way to coarse grain given the information that you have in this case from the crystal structure. Anyway, so obviously a very important thing that one needs to include in any analysis is the calcium binding dynamics, binding, unbinding, because calcium evokes release, and it's known that calcium, if you assume that there is a ring indeed in this structure, pre-calcium, which is not known, it's not established, and maybe it will be established in the next year or so, maybe it won't. But if you do assume that, then calcium is known to disassemble this ring. And that, of course, is the whole idea of this interesting hypothesis from Jim Rothman. So we include, so that's the C2A, the green one, and the black one is the C2B. So there are 3 binding sites to the former and 2 to the latter. And the calcium binding sites for C2B are very close to the interface with the next C2B along. So the thinking is that when calcium comes along, it somehow interferes with that synaptotagmin bond, and that's what precipitates the breaking of the bond. So we build that in. We also build in binding and unbinding kinetics of the snare complex to the synaptotagmin, so they can separate. They'd better be able to do that, or else they're never going to be released. It's known also that the calcium binding loops bury in the membrane when calcium is bound. So these are the various processes that we incorporate. And rate constants are available for pretty much all of this. One interesting factoid that some of you may know is, and it's been a topic of much discussion in the field for a long time, is that the KD value, the equilibrium constant for the calcium sensor, now I forget the numbers now, but it's something like 200 micromolar, which appears to be considerably higher than the concentrations to which this machinery would be subject during release. So people have discussed it, why is that? At first that seems very strange, because it looks like you're hardly going to be able to bind your calcium at all. Why would you do it that way? That's a whole. So I won't say much. I mean, that's something we can discuss. And yet another thing. All of these results arrived the day before yesterday, and they're pretty half-baked. But I thought this is a workshop, and it'd be more fun to present really recent stuff that we've done. So that road to trying to understand and to try to tune to experiment, we've hardly begun on. But most of the results I trust. I mean, it was tremendously bug-ridden. But yeah, I was just making that comment about the dissociation constant because I was talking about these features are built into this analysis. Okay, so we test the washer hypothesis. This is the work mainly of Zach McDarg. So we put this together, this ring with the snare pins and the vesicle and the membrane, and we're going to test it now. So we let go, and we let it equilibrate. We assume it's had ages to equilibrate. The first question is, what does the pre-calcium configuration look like? So there's no calcium there. And one interesting thing is that, none of this is put in. Yeah.
Is this the size of the vesicle? Is it this size? Is it like
Yeah, about 40 nanometers, and this is drawn to scale. Yeah. So in this configuration, here's an almost trivial what we think usefulness of this kind of modeling approach. So the thing is that when you're thinking about all these snares sitting there and this ring and so on, there comes a point, unless you're a mathematical genius with a brilliant internal cinema in your brain, when it's very difficult to see how all the pieces are connected together. For example, it's difficult to see if this configuration is clamped or not in your head. So in a way, just testing the geometry, quite a simple thing. You sit down with a piece of paper and a pen and you draw everything with the right dimensions, is an important issue here. The first question one we would ask, and this is just a question about mechanical equilibrium modulo thermal fluctuation effects. How zippered is this in this state? And it turns out that it's almost fully zippered. So look, there's the linker domain, and there's vamp attached to the vesicle, complex, linker domain, uncomplex. So it's fully zippered down to that point. And what does that mean? So this statement I've written here. So this means that it's this structure anyway is compatible actually with a half-zippered state were that to be imposed by another molecule such as complexin. So there's a lot of talk about how possibly complexin or somebody else imposes a half-zippered state. Suppose this turned out to be 1/4 zippered. Then what is the point of putting complexin, of binding complexin to cover the last half of the zip ring? It's not going to make any difference. But what this says is that it wants to zipper down, and therefore you can imagine peeling it off halfway, having another component come along that peels it off halfway and imposes that half-zippered state. So even that very simple observation I think is quite important. And you may say, well, where is the complexin? Well, it's not there, and it should be there. Nobody knows how it's featuring really. As for everything, there are many proposals out there. And then the next, then the next question is this clamped? So we follow three. I'll follow here three features. The simplest one is just the separation H between the vesicle and the membrane, which is just shown by this line here. And then another important one is the force. So these snares are pulling the vesicle downwards with a certain force. That's the force versus time. That is the membrane separation H versus time. And then the other thing is the membrane energy. That's just the interaction energy. I showed you the various interactions that we incorporate. So this is plotting those energies versus time. And you know with everything you look at there are fluctuations. So what we will embark on in a second is a molecular dynamics simulation that respects the fluctuation dissipation theorem. So it's got temperature in it, and it's got the mobilities of all components and so on. So it's as best as we can do, a realistic dynamics. So is it clamped in this initial configuration? Yes, it is. And that's what, so in other words, if you look at this separation, remember there is a hydration layer here. So what may look like nothing is not nothing. But this is sufficiently displaced that the force is very low. It's of order about a piconewton or something. Now we let the thing go, run an MD simulation, introduce calcium, beginning with what I'll show you here, a relatively simple case of imagine calcium being injected at t equals 0 and then just held constant. The level we've chosen is 50 micromolar. This time profile mimics quite well uncaging experiments. So in uncaging experiments, the calcium uncaging experiments, the calcium level, there's a flash at t equals 0, and then a certain level of calcium, which may be measured fluorescently, is produced, and that falls off very slowly. Has a very long, typically very very long time scale. So we certainly could put in a more realistic time profile, but it would make little difference. 50 micromolar is a bit high. It's certainly studied experimentally, but for the action potential, as far as we know, it's a little bit high. And this is one of the problems. I said that this is somewhat early data. Our N values are a bit low. If we go, the lower the calcium, the slower things happen, right? It just takes longer with a lower calcium concentration to disassemble the synaptotagmin ring. So you have to run those simulations longer. And we are doing that now. So at the moment, we've only gone down to a certain minimum calcium concentration. So what we find is that the entire release process roughly speaking, has 3 stages, which we think is quite interesting. Remember, we're tracking these separation, energy, and force with time. The first stage is unclamping. The calcium comes along, it binds to synaptotagmin monomers, and then after about, for this particular simulation and calcium level, after about 100 microseconds, there's a break in the ring, and that corresponds to the green arrow here. That's a big event. It seems that the ring breaks, the vesicle gets pulled down a bit, and already there's a substantial force which goes up to, I don't know what the mean value is, just to the right of this green arrow, but it looks to be, maybe 20 piconewtons or something. And the energy goes way up at that point. And then disassembly, yeah.
So can you explain, it breaks here because this is the only place where you have 2 calcium bound to the synaptotagmin, or it's a spontaneous break because you had 2 calcium also at other places? I mean, why this particular place? It's because of calcium?
It certainly okay. There is an intrinsic breakage rate which is very, very tiny, and that the corresponding equilibrium constant has been measured. So essentially, what's required is that both calciums need to be bound on the C2b domain, and then an opening is made, which can heal itself or not. So I wish I could answer that question better, but what one needs to do is to look more closely at this particular event. In other words, I'm not sure, how many such breakages up to this point occurred and then re-healed themselves. I don't know. But certainly something happened there. 2 calciums were bound. It broke the connection, and it wasn't able to heal itself.
Yeah, so you could follow that, for example, in your simulations.
Absolutely,
you could have this look at
very,
this.
very important to do that. Yeah, I agree. And I mean, you can see how you can imagine analytical approaches to this. It's pretty easy to calculate at the simplest level. Yeah, how long do you have to wait for a couple of calciums to be bound and so on. But so then the disassembly continues. So for another 50 microseconds later, what's happened is that there are more breaks. And now I've counted this, and you may think this is an implausible statement to make, but I've counted this as the end of disassembly in this little series of snapshots. You may say, but look, it looks to be still stuck. I think that's true. So there's a gray line. There's a gray area rather between this disassembly process and the next stage. Stage 2, the snare complexes, the snare pins as they're called, self-assemble into a ring. And that's kind of happening, it has to be admitted, it's overlapping with the disassembly of the synaptotagmin ring. So there's a different kind of a ring. There's the synaptotagmin ring, which is getting increasingly fragmented. And after about 400 microseconds, what has happened is that these snares, remember they were tangential plus some angle initially, and they've more or less forgotten that orientation, and they spontaneously organize themselves into a ring. It's a stochastic ring. So we're seeing thermal fluctuations here. I've got rid of the synaptotagmin there, so you can see the ring a bit better. We quantify that. The simplest way to quantify that is just by the orientation, the mean orientation of the snare pins, which goes down and down and down, and by this stage has got low enough that we would just about classify that as an assembled ring. And I'll say more about this process a bit later. And then stage 3, fusion. And what is that? Just sits there. Sits there for, in the microscopic world, it just sits there for ages. Just chugs away. The snares have been unclamped. They're released. And now what's happening is that because of this ring and because of its tendency to expand, which I'll discuss a little bit, that tends to pull down the vesicle. It sustains high levels of force. You see, we've gone up to something like 50 piconewtons here. Now, you may say 50 piconewtons, is that big or small? Compared to what? It's actually over a length scale of a couple of nanometers. That's a very big force, remembering that 1 kT is 4 piconewton nanometers. So that's making a big difference to fusion barriers. And we basically just let the system sit there until a fluctuation in the membrane energy hits the fusion threshold, and then we count that as fusion. So of course we're not really fusing the membranes. To really fuse the membranes, you would have to explicitly represent the lipids and so on, and at the moment we're not doing anything like that. But we're assuming the existence of a fusion threshold, and this very, very nice experiments by Fred. He measured something in the range of 25 to 35 kT for his experimental system. It was done by measuring fusion rate, no snares or anything involved, and then doing an Arrhenius plot, and then the slope, compared to the prefactor of 1 over t gives you the energy, the activation energy. So what I've shown you here, after about 1.4 milliseconds, we hit this fusion threshold and we count that as fusion. And we use, for this simulation, if we use an 18 kT fusion threshold, which is lower than that reported by Fred, we get this number, giving a total release time of just under 2 milliseconds for this particular simulation. Now, how to interpret this compared to that? There are two different ways to interpret it. It could be that, let's say that there really are 10 snare pins involved, nobody knows. Then it could be, and let's say we accept this number, and it's very difficult to know if that number should be accepted because the lipid compositions, Fred would be the first to state this, the lipid compositions were not physiological. It's the most important information available at the moment about this issue. But it could be that you get another 7 kT from effects missing from our model. For example, transmembrane domains probably help to catalyze fusion, and many other effects. Calcium is known to have very strong effects on membranes and so on. So maybe there's another 7 kT around there. Or it could be you need more snares than 10. But all we can say at the moment is when we do these simulations without any other assumptions, with 10 snares, it takes this long. With this particular apparatus assumed, the calcium apparatus. So here's all of what I showed you, or similar to what I showed you in a movie. This is the full sequence, but now at a kind of ridiculously high calcium, just so the movie, so we don't have to sit here for 15 minutes. Though David is looking very comfortable in the front row there, so maybe if I gave you some popcorn, you wouldn't mind the full show. But anyway, this is an artificially high. That's obviously the top view. That's the side view, and with time, it's jiggling around. That's those are thermal fluctuations. Obviously, nothing else. Completely passive. And you might say, is this plausible? Is it really that chaotic at the microscopic level? Quite possibly. And when you look at this, it looks like it's taking forever to for the snare pins to release themselves from the. You see how the snare pins have now started to orient. So I better move on because. Time is pressing. But anyway, that is the nature of the microscopic world in terms of fluctuations. That's what KT looks like. And of course, the question is how much is that being restricted by other components which are missing from our model. But we would argue that it is just a hypothesis that the final stage of release involves unfettered snares. So that, it's just a hypothesis, but in the very final step, there's nothing else. So we can take this simulation and we can now predict the kinds of things that are measured in electrophysiological experiments. For example, if we plot the time to the peak release rate as a function of calcium. So each of these calcium values, we've done just n equals 4 runs. That's a lot of simulation time, but it doesn't give us very good statistics, and this n value will go way out with time. And it also, we're limited. We can go down to, as I mentioned, I forget what the lower value, maybe 20 micromole. That should be compared with, say, these measurements in this very well-known paper by Schneggenberger and Neher, Calyx have held. So this is time to peak release. I mean, it's the same quantity, but in a different range of calcium. Actually, this curve is very close to that curve. It's just that these results here are kind of over to the left. But if you extrapolate that, it's close to what we predict here. I think the most I can say is that the model reproduces qualitatively, at least, the kinds of things that are seen in electrophysiological experiments. We can then look at the mean release rate, which is essentially the inverse of the time scale for all of these events to happen. As a function of calcium, the release rate, as you increase calcium, gets higher. And as you get to these to higher calcium values, you start to see a saturation. And that's because there are two things going on here. There's release, there's unclamping, calcium-induced unclamping, and there's fusion. And the fusion rate starts to become limiting at higher calcium. And that's very similar to what's seen experimentally. For example, in these experiments on the retinal, the bipolar retinal neurons by Heidelberger et al. That I showed earlier. We can then, I've described their simulations with constant calcium. We can now, we can do any time dependence we like. So here are action potential runs using a time-dependent calcium profile inferred by Schneggenberger and Neher, not directly measured, but from experimental measurements and filtered through their model. So we use that, and for these particular runs, this is how far we've gone, 1.1 milliseconds. The width of this is about 1 millisecond, so we're probably at the end of the action, but we don't know. Anyway, at this stage, with that action potential, 52 runs, we just have 3 release events. And if that, the error bars are high there, obviously, if you've only got 3 of these release events, but if you take those numbers, that suggests a vesicle release probability of 0.06, compared to, at least for the Calyx have held, about 0.1 is what is claimed experimentally. So what I mean by that statement is that in 49 of the 52 runs, at the end, everything is still intact. And it's got to be that way. As I said earlier in my talk, the vesicle release probability, if you're a vesicle sitting there, most of the time you don't get released. And then for the action potential runs, we can predict various things. This I would not trust at all. We have very poor statistics. We need much better than n equals 4. This is a bit more trustworthy. We can predict the excitatory postsynaptic current of the type that is measured experimentally. So summary, and perhaps we can talk a little bit about, or I can talk a little bit about what the mechanisms involved. So the idea is that what we see happening is that 3 stages: unclamping, then the snares spontaneously assemble themselves into a ring, and then there's fusion. And these last 2 stages are driven by entropic forces. Again, I should emphasize this is not an input to the model. It's not as if we do simulations that invoke entropic forces. There are no entropic forces put in there. It's just interactions between the snare pins. I mean, they can't cross one another, right? It would be insane to have a simulation that didn't include such effects. So what do I mean by that statement? Imagine you start out, say, in this situation. So here's a top view, or that situation, and that's a top view of that situation, with the snare pins completely randomly oriented. then what happens with time if we just let the thing go? And it's not really, that's surprising when you see it happen at all. This happens. With time, they organize themselves into a ring. And that is, quite similar in some ways to disclination effects that are seen in pneumatic liquid crystals. Of course, there you have much, here we have n equals 10, not 6 times 10 to the 23. And that's driven by entropic forces, in the sense that these guys, in the initial configuration, had very little entropy. They're interfering with one another sterically, and that reduces the number of configurations. They don't like that, they do like this. The other, that's the involvement of entropic forces in what we refer to as stage 2. In stage 3, the actual fusion step, what's happening is that so in this little simulation here, the snare pins have been put in a very small ring, artificial initial condition. Now we stand back and let it go. This is what happens. Over time, the ring expands. And this is what's happening during this sequence to the separation and to the force. The force goes way up. It starts down at it starts out at almost nothing and ends up in the 40s. And again, this is an entropic effect. They just have more configurations in this picture than they did initially. What does that have to do with fusion? Well, if the ring expands, because these snare pins are attached to the vesicle, and because of the curvature of the vesicle, it draws the two membranes together, and that's why the force goes up. And those forces are enough to drive fusion. So it's a very different picture to the traditional picture of how fusion is driven by snares. And remember, neurotransmitter release is not, you introduced me as, you said, well, Ben works on snare-mediated fusion. That's not the most important thing. The important thing is, obviously, how does neurotransmitter release happen, and how do other, kinds of exocytotic processes happen. Definitely the snares are a big part of that. They're at the core. But the thinking in the field has always been, and I don't know if, we have a different view. We may be wrong. Anybody may be wrong. I don't think anybody knows. But the traditional view has been that people talk about the zippering energy, and they say that, look, there's so much energy available there. And then they make estimates of how much energy you need to fuse membranes. Most of those estimates, I think, are very questionable, by the way. But that's a whole other discussion. And then they say, look, that's enough energy. Let's say it was 100 kT needed to fuse the membranes. And let's say they're half zippered, so that would correspond to about, from the half zippered to the fully zippered state, you might argue that's 25 kT or something. So you say, I need 4 snare pins. But there's a real problem, in my opinion, or in our opinions, with that argument. There is no known mechanism to transduce that zippering energy into the membranes. Nobody knows how that happens, and nobody has characterized anything at the molecular level which suggests that. I don't think that's what happens. I think they just zipper up. And you see, people resist that because they think, what do you mean zipper up? Why would evolution do it that way? Aren't you just wasting that energy? It looks like you're wasting this huge amount of energy because, you have to, NSF has to prise apart these complexes after all of this business. So, use up lots of ATP. So it looks like a waste, but it's not, because you have to zipper to bring the membranes close, and then they'll fuse if they're close enough. So it's a completely different way of thinking about it. And what these entropic forces mean is that if you have more snare pins, it's clear the movies I showed you. If instead of having 10, I had 15, the forces, the tendency to enlarge the ring would be stronger, and so they'd be pushed together more firmly, and the forces would be greater. And we've previously calculated the fusion time as a function of the number of snare pins. It's roughly exponential. This is not a fundamental expression at all. It's just a fit with a characteristic n star of about 2 or something. And for these particular runs here, you hit millisecond timescales, physiological fusion timescales, with about 15 or 16 snares. So, you know, the subject came up yesterday, I think it was, or the day before, I can't remember. You know, how many snare complexes are required for fusion? And the reported numbers are all over the place. You know, from 2 to 10, and then you make the vesicles a bit bigger. Okay, it's not surprising that has a big effect. This is work from the yarn group. You go up to 30. So, you know, we would say that we argue that what we have found is an explanation of this big range in reported requirements, because there is no requirement. Any number of snares will fuse. It's just that you have more and you'll fuse faster. And different experiments tend to have timescales built into them, which will then pick out a certain apparent number of snares. For example, in assays, small vesicle supported bilayer assays, you have to recruit the T snares laterally. There's a diffusion timescale comes in there, which suggests an apparent number of snares, an apparent requirement. Just a couple of other you know, interesting features that what we have seen seems in our simulation seems to explain. So long ago, the Rothman group showed that if you make the linker, if you insert flexible residues in the linker region, there's a progressive reduction in the rate of fusion. Now, these are called snare pins, and that's because of a kind of a hairpin idea. The idea being that energy is stored, that zippering energy that I claimed was dissipated is actually being stored in bending energy. But then you would expect, if you add a flexible region, that you would instantly abolish fusion. And that doesn't happen. Rather, it goes down progressively, and that's exactly what we see. I'm not showing results here. And another, you know, interesting point worth mentioning is that, you know, people have fretted in the past. I mean, I don't mean to sound dismissive. I mean, it was a reasonable concern. You know, they thought, well, you know, if you zip it down fully, don't the snares get in the way and prevent fusion? No, they don't. We would say they like to expand. And lastly, I'll mention that we believe that these entropic forces among snares are actually important post-fusion as well. Once the fusion pore forms, and I've talked about pre-fusion, you know, 100% today, that's not the end of the story, of course. There's definitely regulation takes place of that fusion pore. There's some involvement of cellular machinery. In these very nice experiments by Erdem Karatikan at Yale, he fuses nanodiscs, these miniature membranes, with cell plasma membranes, and then he's able to, with the patch clamp method, he's able to measure the currents, and so he can back out the sizes of the from the conductances. He can back out the sizes of these little fusion pores, and most importantly, he's able to control the number. This is the beautiful thing. He can control the number of snares. So he's able to see how the size of the fusion pore varies with the number of snares, and these are his experimental results. When you know, comparing 4 snares to 30, for example, you get a much broader distribution. This is an effective free energy landscape. We modeled that using and you'll be pleased to hear, an analytical free energy approach, no simulation whatsoever. And we were able to explain, I think plausibly with reasonable parameters taken from experiment, the dependence, which is very strong, of the fusion pore size on the number of snares that are present. And essentially we say that in this situation, if you have a bunch of snares, so this is a top view of a fusion pore, the more snares you have, the entropic force, just like the rings that I've showed you, have this tendency to expand because of entropic forces. They like to expand, and that expands the pore. So I'd say, you know, this is pretty good evidence that that kind of an effect is taking place, but you know, as usual with these complex systems, it could be 50 other things as well. So let me almost finish with acknowledging the various people involved in this research, Zach and Anaban, and then Satish, Hakamanesh, Jason and Ben in my group. These guys from a while back, and these guys are current. And we've had a terrific collaboration with Jim Rothman and Erdem Karatikan at Yale, and many people in Jim's group and in Erdem's group, particularly Zhen-Yong Wu. And I'd like to acknowledge support from the NIH and also Columbia's internal research initiative, RICE initiative. And if I may shamelessly end with 2 commercials. Is this permitted?
Go ahead.
Yeah. It's not shampoo that I'm going to try and sell you, but or stocks in O'Shaughnessy wine, which exists, but it's not mine, unfortunately. There's a very nice Californian wine, O'Shaughnessy wine. I just quickly an advertisement for postdocs in my group. Anybody, if you know of people who might be interested, we work on neurotransmission, and the other half of what we do is to do with cell division and actomyosin contractility and so on. And also I'd like to mention our department. So I'm in the Department of Chemical Engineering at Columbia, and we are searching for an assistant professor in biology. Very broad search. These are the key broadly defined fields, and perhaps it's this middle one that might resonate with the people here. If you know of any candidates, please encourage them to either contact the department, and they should certainly contact me. Okay, thank you.
Thank you. Okay, so we have time for questions.
Thank you. Very interesting. So it seems to be very important, the curvature of the vesicle for this whole thing to work. So I was wondering, with hormone field, large core secretory granules that are much bigger, like 300 nanometers, do they have enough curvature?
Yeah,
for this thing to work?
that's a great question. So I don't know about that. I mean, I mentioned I invoked in passing experiments the slide with the distribution of snare requirements, if you remember that one. Interesting experiments by the Yarn group, in vitro experiments with liposomes. They investigated the number with liposomes reconstituted with snares. So they found that for the bigger guys of that heading towards that size, there was a much larger requirement. Maybe 30 snares were needed. All kinds of things could be going on. So I give a I mean, I think that's a very good point. And yeah, I think a short answer is I think related things are going on, but they might be qualitatively different. No one knows really.
First of all, excellent talk. Thanks a lot.
That's the preamble to a very difficult question.
No. Absolutely.
No, which I welcome.
Actually, that's all I wanted to say. Excellent talk. I have 2 very naive questions, actually, and I'm sorry if they are too naive. First is the size of vesicle, and is it taken into consideration in terms of mechanical forces which are needed to fuse two membranes? Because compared to proteins, I guess these vesicles are quite
big,
heavy, and they may have much more time to even spontaneously to touch the plasma membrane and to fuse. So, and my second question is about temperature. As far as you mentioned these entropic forces, then it can be excellent testing, testing condition when you change temperature, and can check your assumptions. Thank you.
So yeah, in response, the first question, yeah, but we certainly do. incorporate the as realistic a description as we can of the vesicle. So every component in there, the proteins and the vesicles, has a certain mobility. And then the fluctuation dissipation theorem is invoked. You know, that tells us how the effect of the random forces that represent the temperature should reflect the mobility or lack of mobility, relative lack of mobility in the case of the vesicle. So that's done systematically and quantitatively. And yeah, the vesicle is the biggest guy there, and you're quite right. That's the slowest guy. Oh, sorry. Yeah, part two. Temperature, right. So, no, I think this is an interesting question. I think experimentally there may be a problem, but you're the biologist, you're the expert, because other things may go haywire when you change, in a living system. So in dead systems, if you're a condensed matter physicist or something, yeah, you change the temperature. But I would be concerned that, think of temperature-sensitive mutants and so on, that protein structure may trump everything else. But I think you're absolutely right. That's certainly something, we've looked at that on the computer. It would be very interesting to examine that experimentally, yeah.
So I just wanted to make a comment and a question, or common question. You presented the probability of release finally based on the configuration of the snare and the physical property that you've shown to us. Is it true that if you have a certain calcium concentration, the fact that you have to find the binding site is also something which is a rare event? I mean, you have to go and find the site. So this low probability might be probably a combination of, depending where the channels are located, first of all, the probability for the calcium to find the right place, plus the probability coming, it's not a plus, but it should be accounted for the two different stages. So, because you presented the release probability as just being like depending on this snare.
No, absolutely not.
Okay.
No. That if that was the impression I gave, then I was very misleading. Absolutely not. No.
Okay.
No. It's both, and depending on the calcium level, one is dominant of some aspects, over the other. So yeah, this is the release time, which involves everything from the moment calcium is injected. All of the things you described, David, need to happen. Binding of calcium, and then the synaptotagmin needs to respond, and it may be very robust or it may not be, and then the snares have got to get together and make reach the fusion threshold. Having said that, I know you've studied these issues of rare events. The level at which we treat the calcium kinetics is just it's stochastic, but it's a smeared out mean field version with an on-rate constant and off-rate constant. And it's certainly amenable to refinement in ways that might be necessary. I think you were talking about the very small number of calcium ions that are present and so on. But it's definitely both. Okay. I don't want to overemphasize the snares.
And the second question is, I thought there was still a debate about when the vesicle opens, then the pore could fluctuate or not, and how long the pore stay open. In that particular question, would this snare dancing or pulling be important in the stability of the pore? Have you looked also in simulation in that direction?
Yeah, but that's a whole. Yeah, I think what happens after fusion, that's a whole other complicated story. But fusion pores are seen. They fluctuate like crazy, and actually interesting recent work, Lingang Wu at NIH has looked at release dense core vesicles, you were asking about, that kind of thing, from chromaffin cells. And so he studied the pathway of merger of the vesicle membrane and the plasma membrane in the evolution of the fusion pore. And he's kind of turning the traditional view upside down. The synaptic vesicles are probably different, similar in some ways and different in other ways to everything else because they're so tiny. I mentioned the fusion pore in my last slide, but whether or not, for example, pores actually. We do think snares are involved later. Yeah.
Okay. So, let's thank Ben again.
Okay. Hello, thanks once again, Krasi, for inviting me and David to be here, and we will go from this cellular approach back to the synapse, and I will mainly play on the presynaptic side. We have already heard this morning about the process, how the vesicles come here and get to this state that they can release something. And what I'm interested in more or less is, so what is the transmission, so how is the transmission organized if we look on the organization in the membrane. And in the past we worked a lot on the AMPA receptors and NMDA receptors in my postdoc time, and now in my own lab we work mainly on calcium channels, how they are working on this connection between the vesicle and the channel. So what we have to acknowledge in this chemical synapse is that we have more stochastic processes than guided processes. So we have here the vesicle, and the vesicle can only be released if it has a certain distance or certain neighborhood to a calcium channel, and if this is in the range of 20 nanometer, then the release probability is very high. If it's lower, if it's wider space, then the release probability is very low, and it depends on the kinetic properties of the channel. That depends on the buffering of the calcium inside this compartment, and then also on the pumps that control how many free calcium we have. Then we have this process that glutamate, for instance, goes out and it diffuses between the two membranes, and there we have also a kind of stochastic process that glutamate diffuses and has only a few hundred microseconds the ability to activate a certain population of receptors on the postsynaptic side, and this makes also then the population that we can hit in the postsynaptic side very small in terms of the number of receptors that then can transduce the signal, so that we have here an action potential arriving, and then we have also some transmission of information to the other side. And nevertheless, so the synapses themselves are kind of time coding devices, because they have this ability to decide whether a vesicle goes out or not, and the structures that are behind is always more or less similar, depending on in which kind of species or preparation you look at. So C. elegans or Drosophila, or here in the sensory synapse or the central synapse, you have always these two components that are very similar. You have a cloud of vesicles, you have an active zone, and in this active zone you have a certain region where these vesicles get stabilized, and then you have their calcium channels that somehow important for this kind of process. And what are the common variable between these synaptic structures is that you have a certain efficiency to dock and prime vesicles, and you have then a certain proximity of calcium channels and synaptic vesicles, and that is kind of basic deciding what is your release probability in this particular case. And so what people did in electrophysiology for a long time is to use two exogenous buffers that have a different speed in chelating calcium, and this different speed allows then to control what is the actually spatial arrangement of calcium channels and vesicles. So if you have BAPTA, and BAPTA is, and your preparation is sensitive to BAPTA, then you can say there is a very high a relationship, and if it's not sensitive to HETA, then it's also encountered that, because this is a slow chelator, that these structures have to be very packed and very tight. So you talk about the tight coupling synapse, and the radius between the calcium channel and the vesicle is about 20 nanometer. And then you go to the other extreme, that is a loose coupling synapse, and that is in the range of 100 nanometer. And then both chelators are active and working on. And this means this takes, but this is also, if you then go in the structure of these molecules, that goes close to what is physically feasible in terms of combining these two structures. So you have, of course, a certain space of an ion channel, you have a certain space of a vesicle, you need a certain arrangement. So 20 nanometer is really on the border, but you can imagine. So, and but if you look here in a table that comes out of this review, there you can see that different synapses have different release probability, and if you say the space between vesicle and calcium channel is something important, then there must be also a different arrangement between these synapses. So for instance, if you look here in this olfactory bulb, for instance, then you have a nearly release probability up to 1. And then if you look in the hippocampus, then you have quite a range between 0.1 to 0.8 in this case. And in our case, we use these hippocampal cultures, and it has more or less the same range than in the brain slice what people measured here. So from this, then we can say the preparation that we use kind of resembles the same variability. And what have people done is, in the past, they looked, of course, in using electron microscopy and looking what is the organization of calcium channels in the presynaptic membrane, and they came up with organizations. So here in this very variable synapse, we have kind of clusters of calcium channels. There seems to be non-random. And then if you go in other synapses like the cerebellum, then despite they have a high release probability, there's also a kind of clustered organization. And then they came up even with the idea that they have some structures in clusters, that they have linear clusters, or they have circled clusters, or a kind of star organization. And all this gives you kind of snapshot ideas, because you look only in a given moment where you have fixed your samples, and then you have your freeze preparation. Then you have also to taking into account that your labeling efficiency of the antibody you use, of course, is never being to be 100%. So what you have always a compromise by techniques that you use. And then you have also the other compromise that you maybe have to take, and this was nice from this morning talk to see that you have also this thermal energy in the membrane that puts a lot of different variables in place, which is this, what we believe, this kind of complexes, these molecular complexes that seems to have to be together. They are always disturbed by this energy that is stored in these membranes that agitates the lipids, and the lipids definitely can also agitate the motion of transmembrane proteins and also underlying scaffold proteins or cytoskeleton. And so we can have a steady state rearrangement of this very narrow and tight complexes that are needed to trigger the release of a vesicle. And this is what we are interested in. So what is the impact of this molecular mobility on synaptic transmission, and can we, by looking on this, get a more complete idea what a synapse can really have as a transmission, also a signal transfer. What is the signal transfer of a particular synapse? So what we have to first do, of course, is then to look on calcium channels, and as Fred already mentioned, it's not just easy to go with a calcium channel as a pore that just let in calcium. So the calcium channel is much more than just this kind of membrane ports. It's composed of different subunits, and this is a kind of minimal assembly what you need to have a functional calcium channel that resembles the same kinetic properties as you can see in the neural membrane. So we need an alpha to delta subunit, we need an alpha 1 subunit, we need a beta subunit, and there is, depending on the isoform, also a gamma subunit. All this is, of course, they have to be somehow assembled. these two subunits are important to bring the calcium channel to the surface. Then the alpha 1 subunit is kind of determine what is the flux of calcium, how fast, what is the kinetic properties, but they are also influenced by these two other auxiliary subunits. So therefore, the first aim in the lab was to get a handle on these calcium channels and label these subunits, and we made a lot of effort to get something on the extracellular phase labeled to this pore-forming subunit, and we failed in this badly. So we thought maybe the alpha to delta one would give us a kind of hint where we go. And if you just compare this localization of the alpha to delta 1 in this case and the alpha 1 subunits, you see there's already a huge difference in the distribution of the spots that you can detect. And then if you combine this and get the trajectories, you see already there is a mismatch between this subunit and this subunit, even if you have the co-expression in the one cell. So they don't assemble really. So it seems that in the outer membrane, so this is what we a little bit published in a small publication, but we will go further with this, is that the channel complex is also in itself not a stable organization. So there is of course the pore-forming subunit, but then you have this auxiliary subunits that maybe also go on and off on the channel and maybe tune also a little bit the kinetic of the channel. So therefore, in the end, we went to this kind of construct where we have an N-terminal intracellular tagged channel that has a photoswitchable fluorophore, which allows us then to go in a turf or semi-turf microscopy, where we then can play with this fluorophore in case of photoactivation localization microscopy. So what we do is we can, with a UV laser, we can convert the green fluorophore stochastically to a red fluorophore and then have a readout where we then can easily detect single fluorophores, can fit their position, so can localize their position in place, and then can repeat the cycle, and that we can repeat in a kind of 50 Hertz frequency. And what we then get is this kind of trajectories, and the acquisition rate is of course depending how much photon you can catch from these fluorophores. In the one moment you can see them, and then of course if you want to combine this with mobility, you locked on this one frame to detection. And what we have in the moment is a localization accuracy of about 20 nanometer, 25 nanometer, which allows us then also to go in this small space of a presynaptic membrane to see where is our channel located. And then we went on with analyzing. So you can have this kind of trajectories out of the points that you have detected. Then you can create a coarse grain trajectory. This coarse grain trajectory, of course, will not completely resemble what this molecule might have explored in the moment you look at it, because as I already told you, our acquisition rate is limited. So we can maybe have a different trajectory that's much richer than what we can see. And then we classically go with the mean square displacement already introduced by Doris, and there you can have this either this linear behavior, then you have a diffusion, then you have a hindered diffusion, or you have this curved behavior, then you can say it's a kind of active motion together with fusion. But as you can see here, you need a certain number of points to have a certain precision in your diffusion analysis, and therefore you will lose a lot of the small trajectories that you can see here. And we teamed up with David Olkman in making this kind of step analysis, and this allows us then to analyze much deeper into these trajectories and see also whether in this cloud of trajectories there's not just a curvature, there's maybe also a kind of anisotropy that brings trajectories together or brings molecules together and keep them in a certain space. It's called then this kind of energy wells, and see whether this, and from these energy wells of course then we can extract much more information. So what is the dwell time of a calcium channel in a certain environment, and also what is the size that they explore in this moment. And of course this is very helpful to go into this nano world of the presynaptic synapse, presynaptic membrane. But these are the kind of initial experiments that we did based on this MSD calculation. So what we did is then we looked on PQ type and N type channels. Both of these channels, they behave more or less similar in the membrane. So they have outside synapses, they have a higher mobility than inside synapses. Then we also check this for a couple of other proteins like adhesion proteins, beta and alpha norexins, and also for the snare protein syntaxin, that is also now backed up by a couple of other people that worked on it and ourselves. So that means inside the presynaptic side, we have a lot of dynamics if you just look on the single molecular level, but we have to also admit that the single molecular level is also something that plays an immediate role in the presynapse, because we have not an unlimited number of, for instance, calcium channels. So there is already some experiments where a single calcium channel has been proposed to be sufficient to induce fusion of a vesicle, so by his own action. So that means it is already, I mean, the density is small enough to say, okay, a single molecular approach is something that can also tell us something. And so the question is now, so how to bring this together. So, and the outcome of this first investigation was that the idea, maybe if what we see is maybe there is maybe some confined space, but in principle maybe are stochastic organized, these calcium channels. And of course the calcium channels, they are not just pores, as I told you, they have also kinetic properties. A certain fraction of calcium channels is in an inactivated state or steady state inactivated state, so not all calcium channels that you have in the presynaptic membrane will respond to the stimulus, to the action potential. So there you have only a fraction of the calcium channels, and then if you have a stiff organization and just rely on the kinetic properties, then you might be never can release this kind of vesicle that is indicated here, and you never come close enough that the calcium cloud that you induce is really evoke the transmitter release. But if you have a mobile population of calcium channel, then eventually you have maybe one calcium channel that come close enough and then can release the vesicle. And what it could mean in terms of release probability of a single vesicle, it is tuned, of course, by the density of calcium channel you have available and the mobility that they have. And that could then be kind of a compensation mechanism for kinetic properties that are the channels locked to. So that was our first idea. And so from this now on, we thought maybe there's also a mechanism how to regulate this mobility, because channels are not free floating in the membrane. They have also their confinement, they have their binding partners, they have their crowded environment where they stay in. And a couple of proteins are already well described. It is RIM, this RIM binding protein, also MUNC indirectly in Bassoon. They are participating in the organization of this readily releasable vesicle and the connection to this calcium channel that are inside this membrane. And there's one structure that is quite dominant. That is the distal C-terminus of these calcium channels. And this distal C-terminus has been identified to be to have certain binding sites for these two proteins that seems to be absolutely crucial to organize the calcium channel in proximity to the vesicles that are ready to go. And we looked then in the genetic structure of this calcium channel, and there is a process that is alternative splicing that is due for all the proteins that we have. And there is one splice site that is on the C-terminus, and that evokes that you lose about 150 amino acids, so that you have a short C-terminus or long C-terminus. And this splicing is not something that is just by bioinformatics coming out. It's something that we have indeed in our brains all the time. So we have short versions and long versions, and it means that we have PQ-type channels that have either higher binding affinities to scaffolds or lower affinity to scaffolds. And therefore we thought, yeah, let's check whether these two splice variants that here you can see, so you lose about 150 to 200 amino acids, so it's a sizable structure that you lose. So what is about the proteins? So we made Western blots. So this is expressing in hex cells. So you see the different bands that corresponds to this splice variants, and you see you have these two bands in the cortex and the hippocampus, and what we see is that there's also a developmental regulation of this splice variants, so that you have 50-50 population in young neurons, and then if you go in older tissue, then you see that you have an increase of the long version. So from this on, then we thought, okay, there is some reports that maybe if this is so important, this long version for organization in the synapse, maybe this short one doesn't go really into the synapse. And there might be other options to lock the channel inside the synapse, that is, or this auxiliary beta subunits or the interaction with the snare proteins. But as we have already seen, that the snare proteins are very mobile, maybe this is not the one that links them. And then what we see is that indeed calcium channels, they organize, doesn't matter whether they have a long or short tail, they go into the synapse. And surprisingly, the one that has less binding affinity to the scaffolds is able to accumulate much more scaffold proteins in the synapse. So if we have a small channel or kind of C-terminal small channel, then they seem to enlarge the synapse more than the one that has a long C-terminus. So this match also with recent publications where they have also truncated by demand this C-terminus and see the same thing, that the C-terminus is not really the one that localizes calcium channels in the presynaptic terminal. So from this, we also have here now this confirmation from endogenous synapse that the synapses really go scale up with the number of channels. So if you have in endogenous levels, you have high calcium channel amount, then you have a larger active zone. That is also true for RIM, and it's not true for the snare proteins. That doesn't matter whether you have high or low. The density is inside and outside synapses more or less equal. Yeah. So from this now, we started our experiments and asked, so what is the mobility, the dynamic of these two different splice variants inside the presynaptic site? So we can express here the calcium sensor. This is a GChem synaptophysin together with the calcium channel. So we can control that we have indeed a calcium signal, and then we can have this localization and the dynamics of these calcium channels within this identified synapses. We see that per synapse we have about one of these energy wells that are formed, and from this we can then extract again the numbers. We can have this mobility, size of confinement, dwell time, and there was surprisingly that the dwell time of these calcium channels is not extremely long. It's about 100 milliseconds in this kind of high confined zones. And the size of confinement is about 80 nanometer. And there was one difference that was a little bit surprising, that the short channels are less mobile in these nanodomains than the long one. So that seems that the additional binding sites to the scaffolds are not important for binding. It's rather maybe for steric hindrance, and therefore they toggle maybe a little bit more in these areas. And if we then switch off network activity by blocking postsynaptic glutamate receptors, then we see that the long versions, they get more stabilized, whereas the short one is not really reacting to this kind of manipulation. So that gave us a first hint that the mobility of these calcium channels in the presynaptic site is not just something that is stochastic and we just have to emit as a noise of the system. It's also something that seems to be regulated by additional factors. And so we go then in testing the function of these two splice variants. So we measured calcium in these presynaptic boutons. Of course, we overexpress the channel. We get a little bit more calcium in both. There's not a large difference between the two variants. And then we measured glutamate, and there was a little bit of a surprise again, because the glutamate sensor that we here used gave us the information that in the synapses that we record from and we just stimulate with one action potential, then we see we have a higher glutamate release in the synapses that are overpopulated with the short version, but not for the synapses that are overpopulated with the long version. So that was actually a hint, okay, there might be something different which splice variant you impose. And is also then reflected on the postsynaptic side, that is not just the glutamate that is different, it's also a kind of transmission effect. So even bridging the stochastic process of glutamate release and binding to the receptor. So this is something that could be indeed real and then also functional implication for networks that talk together. Yeah? So that means here the EPSP is larger, though that means we have indeed with this small calcium channel a better release probability than with the long one. And the idea is now, of course we have with both calcium channels we have this kind of nanodomains, and within these nanodomains the calcium channels somehow organized, but it seems to be that maybe this short C-terminus allows the channel to go closer to the vesicle and maybe has a better impact on the release of the vesicle, whereas the calcium channel with a long C-terminus maybe can have a kind of spacing function and is not allowing the channel to go closer to the vesicle than the binding to these scaffold proteins allows them. And so this was one hypothesis, but then you end up a little bit as an experimentalist and not going with some models. Then of course you like to have something that you can prove in experiment. Is there something you can maybe pull? And therefore we went with one idea and say there is now optogenetic tools that we can use for this and ask the question, can we interfere with this dynamic directly and on a defined manner? And this is using this cryptochrome, which is a photoactivatable fluor for a protein from plants. It's also in our body. It's important for the day-night cycle. And these cryptochromes, they have the ability to cluster themselves, so they go oligomerize. And there are some mutants that have this oligomerizing function even better. And these cryptochromes then allow us in a very short time window to cluster the proteins on demand. And they have also the ability to relax over time. So with this kind of manipulation, then what we can do, we can fuse these cryptochromes to the N-terminus of our channels, where we know the channel function is not influenced. So the channel expressed in the membrane, the channel has the same kinetics, the channel is also the same speed, the mobility is not altered if cryptochrome is not switched on. And then what we can see, so this is the situation before, this is the situation after, and then you see there's clearly you can form different kind of domains of calcium channels, and you can come from this diffuse organization of calcium channels in the presynaptic side to a very clustered organization of calcium channels. And then this is here in the broader view, and you see also that this kind of relaxing over time. So in 30 minutes you come back to a normal state, and then you can activate this calcium, so you can cluster these calcium channels again. And so what we then did first is we thought, okay, let's take our glutamate sensor and take these calcium channels that we can fuse together and give a first flash of blue light. So this is kind of our control situation, and then have a certain time lapse in between and look what is happening to the evolution of the glutamate signal that we can record from this particular synapse. And what you can see here, then over time you see there's no evolution in the amplitude, but if you look on this the long tail calcium channel, there we see immediately a difference in the amount of glutamate that comes out, and this decays over time, over the time interval that become longer and longer. And then we were of course positive in terms of, yes, that's cool, and there seems to be some moments that we can interfere with the stochastic motion of these calcium channels, and then we can also interfere with the release probability of this particular synapse. Yeah? And we tested in other experiments where we have then the glutamate sensor here on this side. What is the impact on calcium and on glutamate, or the calcium sensor? So there is no impact on glutamate, but an impact on calcium if we have the short version. And there is an impact on glutamate, but a negative impact on calcium if we have the long version. But this is only true if we have this very fast stimulation. Then we thought maybe if we just cross-link forever, and then we form huge clusters, But what is happening now? Then do we just rule out the difference between the splice variants, or do we have still the same distinction? Yeah? And indeed we have the same distinction, so there's no change in amplitude, but there's a change in the decay, so that indicates maybe you have a delayed release of vesicles. But in this case we have really an onset that is faster because we have a better coupling. So that, but we have in both cases we have a huge increase in calcium. So just more calcium channels, more density of calcium channels doesn't make it sound. So you need still this kind of distance regulation, and if you don't have the distance regulation, then you equal everything. But if you have a distance regulation with short and long C-terminus, then you have the chance to be more precise. And that is also then reflected in the postsynaptic side, and then we ask, what is happening if we now introduce EGDA in our pipette and look what is chelating properties? And we don't see any shift for the short one, but we see a regulation still for the long version. And the idea is now that we come up maybe with the short version is a little bit unrelated to the vesicle because they're losing this binding affinities. But the binding affinities to the scaffold seems to be not very heavy. So there's maybe a low affinity of these interaction sites. And therefore, the long one has this low affinity. And if we then impose another affinity by this cryptochrome, then maybe they assemble in clusters around the vesicles. Whereas the short one, they can't do this. And they assemble because they have this strong cryptochrome binding, so they assemble somewhere as clusters. So they will be similar efficient as they have been before. So they will be tighter coupling maybe. But the long one really can then recruit two vesicles and make a better initial release probability of the vesicle. So this is our idea. So then we of course tested this in our, yeah.
So the experiments with EGTA on the previous slide, how did they enter the picture? I didn't get that. So you have less release with, I don't read it very well, so it's both?
No, if you have crosslink with the long one and then you impose EGDA, then they are EGDA sensitive. So that means they are not as tight as you think. So maybe they're in a distance of 50, 100 nanometer.
Okay, so they form clusters between the calcium channels, but not at
exactly.
the vesicles. Okay, thanks.
And then for the short one is there's no activity of EGDA, so it seems these clusters they are somewhere. Maybe they are even closer to the vesicle, but they are not influenced by this. So if we then go for the crosslink. So this is the situation before, this is the situation after. Indeed, we induce this kind of nanoclusters much more, and this is the quantification for this, so that we can really immobilize channels, and it made us quite confident that the calcium channels in the axonal membrane, they are not just stabilized, so they are always dynamic, they are going back and forth, and we can also recruit calcium channels inside the synapse, so there is a steady state exchange, and if we interfere with this exchange, then we change also release probability of this particular synapse. And exactly this brought us then to maybe a more physiological question to ask, so what is happening if we crosslink and change the release probability? What is in the transfer of information? So how we change then the transfer of information? And this is something as an experiment here. So in the control condition, we give a 5 action potential with a 20 Hertz train, and this is under no crosslink condition, just the tag channel overexpressed. You see there's a little bit higher release, but there is in total there's very random in the amplitude. But then if we crosslink these channels, then we have a very stereotypic picture of the postsynaptic response. We have a high paired pulse, a low paired pulse ratio. So we depress the release of vesicles in continuous stimulation, and this is a kind of quantification, so this is under control condition. Then if you overexpress channels, of course we bring more channels, we modulate a little bit the synapse, as I told you. But still we have a difference between the long one and the short one, which is significant. And then if we cross-link, the short one is not changing dramatically, but the long one changed very strongly, so to a very high-release depressing synapse, which is then also quantified in this picture. So what this brings us in terms of what could be the mechanism is that maybe this long C-terminus that binds directly to the scaffold protein is not to bring the channel closer to the vesicle, but to bring the channel in a certain distance, in a defined distance to the vesicle, and to make the synapse probabilistic. So it's making not the synapse deterministic, that you say, okay, come closer, and then we have always release, but no, keep a certain distance, and we keep maybe a certain also a certain binding affinity, that you're not always locked to this kind of scaffold proteins, and therefore we can use the energy in the membrane as a kind of stochastic moment and can tune the release probability of the synapse. Whereas in the short version, they don't have this kind of connection, they don't have this interference, and therefore, of course, they can place much closer to the vesicle, and they have the better stochastic chance to release a vesicle than in this case. Of course, in what we learned from this looking in the protein expression, it could be the case that we have always a mixture of both, that we have not only long or not only short, but we have maybe a few of them, they come close because they don't have the structure, the other ones they keep a little bit distance, and then it's maybe the mixture within the presynaptic site that makes the composition and makes the synapse different. And a little bit comes this, if you look in this kind of signal transfer ideas based on short-term plasticity, there we have climbing fiber in the cerebellum, parallel fiber in the cerebellum. They have very large difference in short-term plasticity. And then we have this hippocampal synapse that are kind of intermediate properties. And if you set here a threshold and say this is an action potential evoked in the postsynaptic side, then you have here a kind of low-pass filter. In this case, you have a kind of high-pass filter, and in the synapse, in the hippocampal synapse, you have kind of a band filter. And of course, if you then have the properties to shift a little bit the population of calcium channels in terms of splicing, you maybe can set this band pass filter to a different window to be responsive. And another question that we may be addressing now in the future, because we have a mouse model where we can now address this mobility of the calcium channel directly, is to ask the question, maybe in microcircuits where we have combination of inhibitory neurons and excitatory neurons, we have always a problem to decide what is the release probability. And there's an interesting feature that the principal cells, they can connect to different population of interneurons, and astonishingly they have different release probabilities. So they have, on one population of interneurons, they are high release probability, and in another population, on these Matorli cells, they have a low release probability. That means they can really signal different in these networks. And of course, the feedback mechanism of these inhibitory cells are also placed on different space of these excitatory cells. So that means this here we can maybe interfere with the organization of calcium channels. Maybe is also a contribution how this works. And we kind of bluntly tested this by going for different frequency range in our system, so that we have paired recordings from cells. And you see here, this is the control situation. This is with the long tail calcium channel, this with the short tail calcium channel. So this suggests that maybe you have a different population of short and long calcium channels in different boutons of the same cell, and it might be a way to modulate. Of course, there are other ways to modulate this kind of behavior. There are adhesion molecules that play a role in this kind of organization. There's also some other presynaptic molecules that are imposing to this, but what we like to suggest is maybe it's also the splicing of calcium channels that can be additional factor that we have to take in account, and particularly the mobility of these different splice variants. And with this, I'd like to end and mention the people that participate in this work, and Thank you for the attention.
Oh, I will make the first question now that I have the microphone. So very nice. I didn't get how you get much more release with the long channels if they're further away. So do you have more releasable granules for some vesicles for some reasons, or do you have more current with these? Or
yeah, we have several ideas, but we can't, on the post-synaptic side, can't prove this really on a single synapse level. So there could be, one idea is to have multi-vesicular release, so that you have first a single vesicle release, then you assemble more channels around the vesicles, then you maybe have with a one action potential more vesicle release on a given synapse, or you kind of wake up silent synapse. Because if we have a paired recordings, that is not a one-to-one connection in the culture dish. So we have always about 20 synapses per connections, or 10 or something. So we cannot predict how much we have.
But they don't carry more current, these long channels, compared to other ones? No.
In terms of kinetics of the channels, so the short one and the long one, they are identical.
Okay, thanks.
In terms of kinetics, so at least in the heterolog expression system.
So, as far as I can understand, you were suggesting the long ones. The idea might be that they position themselves such that you might or might not get release.
Exactly.
In other words, tuning things so that it's the local release probability for that particular vesicle with that particular calcium channel in proximity is, between 0 and 1, maybe quite. So it seems like then there are, I've always wondered with release, given a certain release probability that's measured experimentally, which is some average number, should one think of every vesicle as being in an identical situation?
I would say no.
But right, so one view would be, every vesicle is in an identical situation, and every vesicle has a certain probability of releasing. So stochastically, you get a certain release probability, or is it kind of, frozen in randomness, to use physicist language, so that different vesicles at a given moment have built-in, release probability. Perhaps you're saying according to the local, the distance for the type of calcium channel and the certain distance. What's your picture? It's a rather sort of general.
I saw your. In your talk you already mentioned this kind of entropy that is playing in because of this stochastic motion in the membrane. I think this comes all together. You have this kind of snare complexes that are many of you. Yeah. Then you have calcium that could many of you, and then it's depending on the stochastic process. So how many snares or how many syntagmines have got the calcium signal? What is the proximity of this calcium channel to this particular vesicle that could be released? And then of course you have also time jitter. And I think with this short one we see already this time jitter. So we have the same amplitude, but we have an increase in the decay of the glutamate signal. That means there may be coming some vesicles later on because they are just a little bit too far away from the direct trigger, but delayed trigger is possible. So, and it would exactly count for this kind of stochastic motions for this very nano-built one vesicle, one calcium signal. Of course, then in some, what we can measure is always an active zone, one synapse. It's not particular vesicle here and there, but it could be exactly this. If you organize it stiff, then it comes better together. If you let it a little bit freedom, then you have a more play.
In your method of tracking channel mobility, do you have any restriction on the size of the synapse? You can do that.
If it's too small, then it becomes a problem.
What is the limiting case?
We have a localization accuracy of 20 nanometers, so I would not go in synapse at about 50 nanometer, so very small synapses, I would say that we have no chance to make a real point. I mean, then we have to have other fluorophores. I mean, one can tune on this to be more precise, but yeah.
So just can you comment on what do we know about the distribution of short versus long calcium channels in endogenous synapses? Are they synapses where you think there could be only one type or a second type? I mean, is it known? What?
Yeah, so what we know so far, I mean, therefore we work on this PQ type channels. So if we would go for maybe for N type channels, we know if we overexpress N type channels, they are dominant, so they will kind of replace the other population of channels that are in the synapses. So in synapses themselves, they are of course heterogeneous in terms of calcium channels. They have not only PQ, they have not only N, they have not only R or whatever type of channel. And all of these channels have a little bit tuning in their kinetics. And in terms of splicing, we have no clue. So we know from this Western blot analysis, the protein is there. So it's not just RNA, but the protein is already there. So you can assume there must be a certain population that could be also in this presynaptic terminals. PQ type channels are not exclusive in this, only in synapses. But so in future experiments, that's what we hope is, because then we can track the endogenous population. That is the aim of this mouse model. And then we can also see if we have success in terms of cross-linking, in terms of faster release, then we know, okay, the population is maybe dominated by long ones. And if you have a synapse that's dominated by short one, maybe the cross-link is not effective. So this is kind of the sensor for the future.
The last question. Is there a way to make the long channels even longer, to actually see how that influences the release probability, and whether essentially the ones that are there sit in sort of sweet spot?
Mm-hmm. So there is actually a disease that is called an epilepsy that is depending on the size of the C-terminus. And this occurs only in humans, so this is a kind of, you have not this kind of disease in rodents, but you can induce this in rodents. And there is actually a group in Japan. They produced a knockout mouse where they kick out the long version. And then they got kind of silent epileptic mice. So it seems to be you need a mix, that is for sure. And in humans you get an additional prolonging of this C-terminus, and it has the same effect. It makes you seizures. So you get the problem if you prolong the C-terminus even further. So this is a polyglutamate stretch that you add on it. So it's a inherited disease. It's very few cases, but it has kind of negative impact.
Thanks.
Okay, let's thank Martin again. So we are going to close this morning session, and we are going to start again at 2 o'clock. Not 3, but 2 o'clock. Okay. See you this afternoon.
So, I should introduce David. He will be the next one, talking about, hopefully, the theoretical part of the analysis I used, and maybe some others. And yeah, we are all there. Let's go.
Okay. Okay, so first of all, I would like to thank all of you for coming and really enriching from questions, from thinking, comments, and your talk. I mean, our understanding of these micro nanodomains, calcium, receptor trafficking, the role of the ER, and more. And so today, I would like to bridge between presynaptic terminal and the postsynaptic terminal. So in between these two spaces. So first, to explain the analysis that Martin didn't have time to show in detail, how we analyze single particle trajectories. And I want to move on to nanodomains in the postsynaptic terminal. It's really an advertisement for the next talk, which will be given by my PhD student Kanishka. So how do we basically analyze calcium nanodomains. So first of all, I would like to dedicate this talk and this conference in general to my former collaborator Zev Schuss, who just passed away this year. And so if you are interested, I posted an obituary on my webpage about his life. We started as a chief orchestra conductor and move on slowly to apply math, to biophysics, and his path is actually revealed by his own trajectory as a child across the previous Soviet Union. It's called the Tehran Children. This is a 3 years story of children during World War II, escaping from Poland, moving to the east until Novosibirsk, and coming back here to Palestine at that time and Israel now. And so if you're interested in this story and his contribution to applied maths, you're welcome to have a look at this obituary that I'm not trying to get published in a journal. And this is another story. Calcium nanodomain in the pre- and postsynaptic terminals. Let's start with the pre. And so this figure has been introduced several times before, where we are interested in how calcium can be restricted after flowing through this calcium channel in order to activate these vesicles. And we have heard this morning that these calcium channels are not necessarily fixed, but they can move and can be trapped. So I would like to discuss this. And also, I'm not going to go to the step that Ben has described, that is, once the calcium are bound to this specific synaptotagmin, they do all kind of physical process here that he described are involved to create a fusion. So I'm not going to describe this, but I'm interested in what happened from the moment calcium enters to the moment that calcium binds. So this will be the first part. And the second part, I'll explain that because these channels are not stable, how do we analyze this single particle trajectory that Martin presented just the talk before? So, in particular, how can we be quantitative about the effect or the impact of the distance between the calcium channels, this specific location, assuming it is fixed, to the position where the synaptotagmin can be located underneath the vesicle? So you could find, you can see in the literature that you get graph of this type, that is, as a function of the distance, there are estimates about what is the concentration of calcium after it enters through these channels. And so I have been puzzled about how can you get such an estimate, how can you get such a result? Because the measurement at this scale is very poor. So most of the work in that direction has been obtained through modeling and simulation. Now we have to question what is the correct or what is the appropriate environment of the presynaptic terminal that we should put in order to get this. For example, is it sufficient to have something in 2D or 3D with an empty space type picture? Or shall we put all the obstacles that are created by those vesicles? So again, what has been published in the past years is mostly 2-dimension, or 3-dimension is really ignoring the 3-dimension. Everything is like 2D in terms of calculating this distribution probability. And indeed, people have started to look at what happens if you have a uniform distribution of channels, what happens if channels are clustered, and trying to get information about how this is important, as Martin has already started to explain, in a much more refined detail, because it was through the super resolution trajectories. So this was before. And by fitting data, trying to connect calcium concentration to the vesicular probability, empirically, people came out with number as a slope, giving a sort of connection between the number of needed calcium channels, calcium ions, to the probability of release for different type of synapses. So I would like to revisit basically this concept. So how the three-dimensional geometry of vesicle matters. Does it really matter to have here a sort of obstacles in this process of a calcium ion after it flows to find here this red part, which I consider as an absorbing part? When an ion enters here, assuming that with probability 1 it will bind to the binding site of synaptotagmin. So how do you address the question of the shape, and in particular how can we use this to calculate and simulate the probability that this event happened compared to the event that an ion entered and it is lost in the bulk? Because if the ion is lost in the bulk, the time it takes for an ion to come back here is going to be much longer, although it can be important, compared to the time it takes when this channel is very close here to go directly and enter into this region. So for this, we created a Brownian simulation where we started to investigate how long it takes for a Brownian particle when, for example, it started in the bulk to find here this narrow red ribbon. So let me enter a little bit into the math. So what we did is we started to, because of the symmetry of revolution, you can slice the domain and study basically everything in this two-dimensional domain, taking into account the symmetry of revolution. And so to calculate the time it takes for this to happen, we can solve what is called the Dynkin equation, which is Laplacian u equal -1. So u represent the time it takes for a single particle to find this ribbon. You put 0 absorbing boundary condition on the ribbon, and du dn, that is reflecting boundary, the particle is not, or the time associated to the particle is reflected everywhere else, and you are interested in what is this time as a function of the size of the ribbon. So we solved this problem basically by brute force, using what is called conformal mapping in dimension 2, and we mapped this Laplacian equation into the conformal Laplacian, where the conformal mapping is 1 over xi. And this was done by my previous student, Claire Guerrier, who's moved to a postdoc. And basically, you have now to solve a partial differential equation in this new variable, and this domain is transformed into a very long strip. And now, because of its a long strip, you can approximate it as a one-dimensional problem. Yes?
Excuse me, David. Why the omega prime domain has to also be like a ball or sphere? Like
no, it doesn't have to. It's more convenient here. Because then you can map circle into lines. But it's not important.
Because it seems like around your target zone.
It's true. It has a little effect, but we are going to play with R2 later on. So R2 is very big. It's almost flat.
Okay, all right.
So it's not that crucial.
What was epsilon?
Epsilon is the width or the size here of this small ribbon. It's where your snare would be basically located in this ring.
So that's a small parameter.
That's a small parameter. Yeah. And you want to see, as this small parameter goes to 0, the time basically will go to infinity, and the question is how it goes to infinity, and this will give us the formula that we want to have for this problem. Because
when you were running your simulations, did you get out the distributions as well? Because
did you what?
The distributions of the first passage times, because now you're working out the mean first passage time.
Yeah, the distribution is, at the time scale we are interested in, is Poissonian. So you are just interested in the first exponent, because basically for this stage here, at this stage, we are interested in what happened when you start in the bulk. Okay. When you start in the boundary layer, it's another story. I will come back to that, where you have to look at the probability, and then we'll get a specific spatial dependency.
Okay, so essentially interested on the tail.
You're interested in the tail because it is long time. When you start here, wherever you start outside here, the short time doesn't matter. So I will go quickly on this, but basically you can expand your solution as a function of the parameter epsilon or epsilon star. You can then separate your equation according to this expansion and look at the first term in this expansion. You can also look at all the terms, but basically y0, which was the first one, was capturing already the information we wanted, so that ultimately the solution here depends on a constant a, and s is the distance from in this conformal map is the distance between the hole and a point on this band. And so what you get ultimately is that the time it takes when you come back to the real space depends on the volume. 4 pi d is the diffusion coefficient, epsilon is the size, r is the radius of the vesicle, and we have now the dependency as a function of the position. And so again, I want to emphasize this. The surface of the ribbon is epsilon to the power 3/2, but the mean first passage time is 1 over epsilon. Okay, so then we now have the time it takes to go from here to this ribbon, and now we were interested in a different question. Now suppose this distance here can vary. What's the probability for a Brownian particle, like for calcium, to hit this ribbon before being lost? So now we have to define what is the meaning of being lost. So for this, you can choose an arbitrary distance here, height, from the plasma membrane. And indeed, as soon as you are at half of the ball or after the entire vesicle, you can consider the particle is lost. It's going to be very hard. It will take a long time for this Brownian particle to come back here. And so this is what is said here. The time it takes when you're outside, it takes basically seconds. The time scale it takes when a calcium ion has missed the target, then it takes seconds by Brownian motion to go back. That's why the short time asymptotics of the time is unimportant. So to calculate this, we looked at the classical splitting probability. This is extremely well-known equation. You put Laplacian p equal 0 in the square domain, and p equals 1 in the part here you are interested in, reflecting boundary condition here, and p equals 0 on this upper part. And you can calculate actually this probability explicitly as a function of the distance r to the channel, that is as a function of the distance between where you inject, which is your calcium channel, and this ribbon, between the distance between these two objects. And so we get a specific expression, analytical expression, that depends on this h, but you see when h is very large, these terms basically drops, and what is interesting is this term here. Otherwise, you have some correction here to be added. R is the size, the radius of the vesicle, and that's it. Note that it is independent of the diffusion coefficient. Whatever the diffusion, this is unimportant. And so we use this, because this was analytical, to predict what is going on, the probability to activate a vesicle. So I'll explain what we meant by a vesicle being activated as a function of the distance. So we said, let's suppose that you need at least 3 ions to come to this ribbon. You see, 2 can be located at the synaptotagmin, but you can have, if it's more complicated, you can have severals, but there is a combinatorics here to be done, because you can have 1 per synaptotagmin, but not necessarily 2 on the same one. So what happened if you have 3, 4, and 5? And we started with a different calcium ion entering. What happened when you have 10, 100, 500? And you can see the very sharp decrease in the probability at basically 20 to 40 nanometer. Of course, this sharp decay depends on the amount of entering ions. If you have a lot, as you can see here in dark, it's going to be different compared to the case where you have a very small amount. But the spatial scales is few tenths of nanometer, 20 to 40 basically. So we calculate this probability as saying that we calculated this curve as saying that you need to have a threshold. Once you have more than 2 or 3 ions, then what is this probability? And you can calculate this as a function of p that I just calculated in the previous slide. And then you can explore what happened when you look. Suppose you want to achieve a given probability. Let's say you want a probability of 0.8. And now you can have a graph. How shall we put a given number of ions and at which distance to satisfy this condition? And so this gives a specific restriction in this 2-parameter space as the function of the position and the number. And so we looked at this this is basically 2 types of vesicular configurations. So now the next question we wanted to ask is that suppose now you have a grid of vesicles. You can vary the location of the vesicles, and you assume that you put the source of calcium at a specific point. What is going to be the distribution of calcium going into these 4 neighboring vesicles according to the position? So if you are very close to this to the blue one, basically everything will go here. But as you move away in this direction, you will see that then you can start splitting into the different other vesicles. And this again will have some impact, because if you have one ion bond to synaptotagmin based on different vesicle, it's not the same that having many ions bond at the same vesicle for this probability of release. And so then we wanted to make a model to account for the calcium that goes inside the calcium and say what happened to calcium. A certain fraction of calcium, so this is not to answer basically Ben's question about are they prepared vesicle to be released, and can we address this in terms of calcium already preloaded at the specific site. So if you inject a calcium flux, a certain fraction will go to the bulk. They are lost. And then we treated them as mass action law. That is, they can bind, calcium can bind to buffer can be released, and this is the classical theory of chemical reaction in the bulk where you don't distinguish any more individuals. The calcium can be pumped out, or they can be extruded with a certain rate. Or what can happen to calcium? They can go underneath the vesicle and bind to a calcium buffer underneath, or synaptotagmin. And then we made here a sort of we made a Markov chain here to account for this counting process. You count how many ions are bound, and then of course we can use forward the backward binding rate, saying sometimes we can in the model explore what happens when there is no binding rate, that is the binding rate, unbinding rate is infinite, or you have a given finite binding rate. Okay, so there are 2 fates. And what can happen at any moment of time, a calcium ion that is in the bulk can by chance, because if there are many ions, they are all not been buffered, by chance some of them can come to the right location. And this will be important for what we're going to see for a process called asynchronous release. That we think we have some kind of modeling or possible explanation. At least we have some offer about this. So we can write the equation about this transition probability. This is the probability that you start with 0 synaptotagmin bound, and then you have 1, 2, and 3. So you have each time you have a flux either from a calcium ion coming from a channel that can increase this probability, or you have one of the free calcium ion, this is the meaning of the second term, that can come and bind underneath the vesicle. And so you have to couple this set of Markov chains to the mass action law, so that we have a coupled system of mass action to this Markov chain equation. So again, this is a classical number of free ions. So you can have unbinding from bound ones, and so you move on with this is a classical term. And then this is what happened. You can have, for example, after a vesicle is released, and if some ions were there, they can be released back into the bulk. And so what we had here is a term with a given threshold, saying that how many ions you have to accumulate to a certain threshold T that you can fix in advance for a vesicle, for example, to be released. So with this, we simulated, for example, a single pulse going through a given channel. And now this is the time of the vesicular release according to a threshold. So I think we put threshold equals 3 and 4, and we varied this. So either you have 0 buffer in the bulk, and then we added buffer. And now we ask at time 0, you give this pulse. What do you see in terms of the distribution of vesicular release? First of all, there is a big high probability of release, which is this first peak that we are seeing. However, you see that if there are no buffers, you see a distribution of release time. And as the buffer concentration is increasing here, this second peak disappears. And so then, looking at the simulation, we understood that this is due to the calcium ions that are in the bulk and that coming back underneath the vesicle at a time scale that can last hundreds of milliseconds and could be responsible for asynchronous release. And this is a population. So for example, you can have a vesicles that immediately after the calcium enters, it will have all of this binding site, and then we assume with probability 1, we didn't account for the synaptotag mean probability in this model. It's going to be released is content, but what can happen is that you can have vesicles that are preloaded. That is, for example, they have, suppose the threshold is 5, and they are preloaded with 3 calcium. Then you need 2 calcium to come during this time to this specific location for this vesicle to be released. So that's why this suggests that you can have preloaded vesicles that are waiting basically to be released, and they are released because of this stochastic event coming from the bulk in this very low buffer condition. If you have very high buffer condition, this will not exist. So the idea is that you will have something which is not showing any kind of asynchronous release.
David, when you say preloaded, you mean from how long ago?
For the first pulse. So you wait.
Okay, from the first pulse,
that's
preloaded.
Right.
Okay.
That's right.
So in other words, because it had better be, you know, if you wait long enough, they're going to dissociate, right?
That's right. Yeah. That's right. So we played in this with this constant. So this is exactly what. So just to summarize, you can have this entrance of calcium. I mean, there are some specific type of synapse where you have this ribone where vesicles are going underneath this additional structure, which play an important role, especially at photoreceptor synapses and many sensory synapses cells. So we have this Markov chain, and now we looked exactly in the question you just asked. You can have vesicle that docked, undocked, and in addition you can have a binding site that could be important. And so I just want to finish this part looking at this graph to try to explore what could be synaptic depression or synaptic facilitation according to this scheme. synaptic depression is thought about, you don't have any more vesicles available. And so you can see that you get, for example, if you have, after a vesicle is docked, no other vesicle is coming to replenish, you start to see that you go from a certain distribution to a lower one, showing this depression type activity. If the docking time is immediate, as soon as the vesicle has been exocytosed, another one comes immediately. And so with this calcium effect, you can see this facilitation, that is, you have an increase in the release probability. And the reason for this, so this is in between two spikes, I think it was hundreds of milliseconds. And so the idea is that the vesicles are preloaded. And they are preloaded, although it's not, this is usually inaccessible to do this experiment and trying to understand what is the concentration from an experimental point of view in this nano domains. So the simulation suggested that this increased in this in the facilitation is due to this preloaded calcium on this binding site. So it didn't change much if we had fast versus slow unbinding with this time constant for the fast and slow. However, if you add here this effect of vesicle not being replenished, we saw here that we started to see this depression effect due to vesicles. So in general, I mean, we are now working on having more spikes to understand how depression and facilitations together can be explained through this calcium accumulation underneath the vesicle and the time scale of the vesicle for the vesicle to dock to the right place. Okay, so now I would like to move on from this analysis of this calcium nanodomain, which is completely static, to what we have heard this morning about the calcium channels. Actually, they are not necessarily fixed, but they are moving, although Martin started to show that there are regions where this channel could spend or can be trapped, transiently trapped, for a time scale could be long enough, hundreds of milliseconds, so that they are at the right place at the right moment. So many years ago, Martin gave us some of his trajectories from which we could extract, we could see specific accumulation. That is, the trajectories were concentrated at specific places. This is a density, this is a diffusion coefficient. And when we calculated at each point, so you do a specific analysis here, you don't average over a trajectory. You take a specific spot and you calculate the velocity using all trajectory going through this spot. It's a different way to calculate velocity or to calculate the diffusion coefficient. And what we could observe, those are all the data that have moved on after the years, but basically we could find that there are regions of high density, and this region of high density, we calculated a number, which I'm not going to describe here in detail, that told us how these places can be represented as potential wells, as harmonic potential wells. And so what's the idea behind this is that you remember the new second law of motion from Newton, force equal acceleration. Langevin modified it, adding the noise term and the damping term, and Smoluchowski more than 100 years ago made the following analysis. When you are in an overdamped medium, you can basically drop the acceleration so that the velocity plus the force is basically equal to this, another way to say it is that the velocity is driven by the force plus Brownian motion. And so over the years, we have developed tools more and more sophisticated to, can you extract the drift and the diffusion coefficient or the diffusion tensor from trajectories? How do you do this? And so basically, how can you identify a feature? So here, what you see here, you see many of these small drift computed from trajectories. Can you find here a feature, pattern, that can emerge. And so basically, we focused on the region of high density and said, can a region of high density be defined by the gradient of a conservative potential that will restrict the position? And so this is a classical picture where you have a potential well, and you're in dimension 2, you have this parabolic well, and if you calculate the gradient, what you see is velocity that should point out toward a single point. And this can be considered as a signature of this potential well. And so as Martin showed this morning, analysis of his trajectories show that this region of high density are actually correlated very highly. We have a score to compute this to a potential well. And so from this potential well, we develop algorithm to calculate precisely the location of the ellipse and, in addition, the depth of the potential that will give us altogether an estimation of the energy, of some energy which is something abstract here, because this is not driven by any classical physical force like electrostatic. We're talking here about a size of, in average, of 18 nanometer. So how can you have a force that extends to such a domain if you think about just electrostatic? It cannot be. So to that day, and I'm going to show that it seems to be universal, what we have described here. We have no idea what is the exact mechanism, the physical mechanism of this. And so Martin already presented this. So we looked at basically the size for different short and long tail. So Martin has already introduced to you this morning the difference between the two and the cry when they can be cross-linked. So to summarize, we found a size of about 80 nanometer. The diffusion coefficient is shown here, where we have some variability. And the residence times was between 100 and 200 milliseconds. While the energy of the well was in average, between 3 something, which is a very small amount of energy. But probably this is what we need after a vesicle has fused to have this reorganization. You don't want to trap the calcium channel forever. You want them to be relocated at another place after a vesicular release. And so, so this, we have seen already this morning that the place where there was this high calcium, the place where we had this calcium channel, were also the place where we had this calcium activity, showing that these places are important, at least in a correlative manner, to the place where calcium is important for the cells. So at the presynaptic terminal, and there's marker now that's to make sure that we are at the synaptic terminal using synaptotagmin. And basically, there was no statistical correlation between the size, the energy, and the diffusion coefficient, as shown here by those plots. That is, those parameters seems to be independent. In other words, you can have a size of a potential well that is not correlated with the inside diffusion coefficient or the energy. Although they participate, but they are here, as shown, independent. One interesting aspect is you can ask how stable are these potential wells. The receptor here can transiently move and stay trapped for hundreds of milliseconds. But the structure themselves, how long can they be stable? So according to this analysis, doing this time step analysis where you use between this and 20 milliseconds a completely different data, but not in between 10 milliseconds, but from here to, you have completely different set of data, and yet with these different sets of data, you can reconstruct this nanostructure. And we found that the average time of this structure is about 30 seconds. Okay, is calcium dynamic exceptional? So we looked in the past 10 years about statistical analysis of many single particle trajectories, such as the GAG protein for HIV virus. We looked at AMPA receptors. We looked at calreticulin from the endoplasmic reticulum, or this calcium voltage channel 2.1. In all cases, there are regions of high density, suggesting that this is a general mechanism, although the exact mechanism we do not understand, but there is a general principle of clustering receptor at specific locations. And indeed, let me remind you that for postsynaptic AMPA receptors, many years ago, this is where we first started all of this analysis, we could find potential wells associated to region of high density of these AMPA receptors, and these potential wells were exactly located, at least for a large fraction of them, on the postsynaptic density. And at that time, I mean a couple of years after, people looked at the same structure and found the same structure for these glycine receptors. Again, we could find at that time that AMPA receptor domains could be stable for hours, while some of them could disappear in 15 minutes. So there were more stable AMPA receptor nanodomain, which were much more stable than these calcium voltage channels nanodomain. And what was interesting at that time is that for these AMPA receptors, although we had at that time much fewer points, but the energy was bigger, and the size of this region were of the order of 300 nanometer, not 80, much bigger size. So I would like to put here in parallel, to conclude this part, that the high density region in this postsynaptic terminal, or this potential well associated to this AMPA receptor, were of the order of 200 to 40 nanometer, with a diffusion coefficient which was ranging from 0.03 to 0.2. They were stable over tens of minutes. The residence time were tenths of seconds compared to milliseconds. And the energy could range from 3 to 6, with some cases of energy less, more than 8. High density region in the presynaptic terminal, instead of going into the range from 200 to 400, they were mostly peaked at around 80 nanometer, with a diffusion coefficient here. I didn't show you the variance, but of the order of 0.05. The stability was about 30 seconds and not tens of minutes. And the residence time was about hundreds of milliseconds. And I want to insist that this hundreds of milliseconds, if you do just a calculation or simulation for the same territory, just using Brownian simulation, by Brownian motion alone, we have a 4 to 5 times longer. That is, the potential well maintains the receptor at this place. And the energy was of the order of 3 to 4. So I wanted to show, as a result of probably our 10 years in this analysis, to show the difference between pre and post. Now I would like to connect, yeah? No. You cannot do that, because so the question is about the residence time. The residence time, people have calculated residence time when they had quantum dots, and estimated because they could have long trajectories. Here we have short trajectories. So here there is some analysis to first pre-calculate the potential well. From the potential well, you estimate the energy, and then you use a classical activation theory to estimate the time. You don't calculate any of this through trajectory. This is the beauty of the method. That is, you reconstruct this energy information, this energy landscape, or this difference of energy, from which you do this calculation.
Yeah, so it's a computer. The resonance time is computed. Sure. It's not observed.
Absolutely.
Okay. So, but how can you show that this is correct then, the resonance?
No, I'm not sure. I just show you the result of the analysis, and we hope that this gives a good estimate, because we believe that the physical theory is okay. Okay, so now I would like to discuss, to introduce the next talk, an experiment that Eddy Korkotian made. So I need your full attention now to explain the experiments. Okay, so let's start, and Eddy can intervene at any moment of time. So what Eddy did, and we could not understand these experiments I mean, I could not understood for years until we understood the logic, but it took us years. Let me explain. Eddie does the following uncaging experiments. He, at time t equals 0, you free calcium in the head, and you measure calcium concentration in the head, or numbers of calcium particles, or the fluorescence, and you compare it with what you get in the dendrite. Time 0, you uncage, and then you record. So you uncage, and you see what happened in blue in the head, so you have the decay of the calcium, and at the dendrite, here, you see this increase. If you repeat the same experiments, and here we're talking about a structure that Eddie has already introduced. It's called the spine apparatus. What you don't, so he has a marker, I'll show it in a second. When you don't have spine apparatus, there is basically nothing. And now, to show the asymmetry, if you encage here or encage here with and without, you have basically more or less the same phenomena. So the experiments say that there is an increase in few milliseconds of calcium from here to here. So then I asked Kanishka, maybe we did wrong in all our simulation for the past 10 years. Can you repeat the simulation I started 20 years ago? Just encage at the center, calcium, and please tell me how long it takes to arrive here. Because I lost, at some point, confidence in what I did myself. So he did this, but if you look at the scale here, this is 1, 2, 3, 5 calcium. So this was at the time scale of 10, 15 milliseconds, you get nothing. That is, this should be, if we didn't put at the scale, this is flat. So they said that, first of all, this wrinkle myself, that indeed, at this such a time scale, how come you can have such amount of calcium already in the spine, in the dendritic spine, in the dendrite? So to confirm this, and at that time, many years ago, Menachem and Eddie did this experiment, and it has been redone over the years by many groups, and this is the latest one with STED microscopy. But whatever the experimental technique, the timescales of this recovery or this FRAP experiment, when you look afterwards at the calcium at the dendrite when it is initiated in the head, is hundreds of milliseconds. And this is consistent with the simulation we did many years ago. And also it is consistent with the analysis we did over the years, where we could calculate the mean first passage time it takes to go from here to here. It depends on the volume, on the radius here, on A is the size of the head, there is a logarithmic term, there is L square, which is, L is the length of the spine neck, and there is a term here, V multiplied by L, which accounts for the trajectory that can come here and can return an infinite number of times. And it takes, indeed, hundreds of milliseconds. So this is how everything started a couple of years ago, and we had no idea. We didn't understand, because specifically, we needed to find a messenger, something that will propagate quickly at the order of milliseconds. And diffusion alone seems to be not possible. Either if it has to activate, and if it has to activate pumps and channels, it's too long. It has to, even if it went into the ER or outside the ER, it is too long. So what was the mechanism? So I don't want to say too much. I want to introduce what Kanishka is going to reveal and to show specifically how he did the simulation. I just want to give you the answer to the question I'm asking. And for this, to remind you that this structure, which is called the spine apparatus, on the surface of which, so it has been already introduced, you have ryanodine receptor, IP3 receptor, circuit pump, and many others. So can this help us to understand the fast time scale. Is there something here that we can use to understand this? Again, if you think about binding, it takes time to bind. And so it was still unclear. So what we did at that time, I said to Kanishka, why don't we make a simulation and see what happened? A Brownian simulations. So again, you have this structure, and again, we thought, as Eddie pointed out, this structure may occupy so much space that it can impede, it can prevent calcium actually to go physically from the spine head to the dendrite. And so now we were really trying to understand how the cell is organized at this nanometer or tens of nanometer scale. And is this important in simulation where you don't look at the specific distribution of receptors? You disregard this geometry, and in most of the case, that's okay. It doesn't matter because you have enough calcium, everything is sufficiently homogeneous. But here we have this very constraint of the geometry. So again, this is a figure given by Eddie showing that specifically you have a marker of this spine apparatus showing that indeed this structure is exactly located between the head and the dendrite. And so Kanishka started to do some simulation, putting circopumps, ryanodine receptors, and he did some simulation, and he will explain in his talk how he did the exact simulation. But one thing came out of the simulation is that if ryanodine receptors are closed, you get nothing at the base. But when ryanodine receptor could be activated and ryanodine receptor could release ions, we started to see something that became significant. Yet, the question is, what was the time scale? Why 3 milliseconds? Why 5 milliseconds if it has to do with diffusion? So let me reveal the secret, and then Kanishka will explain the simulation and all what he found about the structural organization of these receptors. So where the calcium in the dendrite is coming from, is it the same calcium that were there that we could find at the dendrite, or is the calcium coming from somewhere else? And what is the mechanism underlying a so fast time scale? So it took us some time, but then we realized that probably what we did in the last 10 years could not be applied this way about calculating mean first passage time, narrow escape. What we thought is that finally what is needed to have such fast time scales, first of all, we thought about electrodiffusion, that maybe there is a drift that pushed everything, but it was not that clear, that because we could reproduce here with this simple simulation the result. And what we started to understand is that probably the mechanism of fast signaling has to do with the first ion that arrives. And this is known in other fields, although there was very little discussion in the biology literature about extreme statistics. That is, you have many ions, but ultimately, it's not that important to have so many ions. What is important is to have the first one that have to travel a certain distance away to activate the receptors. And if you have a mechanism of amplification, that's it. You don't need all the ions to be transported from the initial part, I mean, for the place where you have this increase, to the place where you need to have a second an amplification or a second pass, a second step in this process. And this is exactly what we are used to if you think about extreme statistics of running marathons. What you are interested in, what people are interested in, is not usually about this distribution, is how long, what is the time scale of the first one to arrive. That's it. If you look at TV, on the TV, on the media, this is what you are basically spotting. Oh, the guy did such amount of time. That's it. And so then this is exactly what we looked at. You say, what is the time scale for the first ion to arrive? And so we found that we obtained a distribution. So this analysis depends on the numbers I'm going to show in a second. And Kanishka will explain to you, we also looked at the distribution of the second ion to arrive. And indeed, for the first one and the second one, we could find an average time scale of milliseconds, which was exactly the time scale we were interested in for this process. Now, let me explain. So we started, so we believed in the simulation, but we wanted to have something more deep in terms of the connection with the parameter. So we said, suppose we have n random walk. What is the mean first passage time for the first one to arrive? And actually, this mean first passage time is the distribution of the random walk if you have one guy to the power n. And this probability, the survival probability, is connected to the integral of the probability distribution function, which is diffusion equation, where everything is reflecting, and here you have absorption. And the distribution for the first one to arrive, this is the flux, multiplied for the flux for the first one to arrive, multiplied by the probability that n minus 1 ion are still inside the domain. And so from this, we develop, so in dimension 1, there was some work. It was it was worked out. That is, suppose you are releasing at a certain distance, let's say at the origin 0, in the interval minus infinity to a, you have n particles. How long it takes for the first one to arrive? So here you can calculate everything explicitly. And the short time asymptotic, so this is basically the mean first passage time, you have to calculate an integral. And by moving on with the classical change of variable, I mean, not that classical, you find out that the time it takes depends on the distance divided by the diffusion coefficient and divided by the log of the number of particles. And this is, you can also do the same analysis if you have a finite interval. And what we develop is a calculation to do this in 2 and 3 dimensions. And just let's go to see what happened in 2 dimensions. Or 3 dimensions, although the mean first passage time, let me remind you that dependent on the entire volume and the diffusion coefficient, and A is the size here of the small hole. The time it takes for the first one to arrive has nothing to do with this geometry. It has nothing to do. The particle, the first one, shoots toward the target. So you have, instead of having the volume here or the surface, you have the distance, the Euclidean distance, the geometrical distance, or also the geodesic. If you have an obstacle, we're going to see that in a second. And it depends. There is some geometrical feature. It's not anymore log here. You need some geometry, which is the log of epsilon. Epsilon is the ratio of the absorbing to the total boundary. And in dimension 2, it's even more surprising. There is a square root coming out. And that depends on the distance between the source and the absorbing window and the size of the target. It's different geometry, suggesting that, for example, all the analysis of using that homogenization to calculate diffusion does not make sense anymore. You don't calculate anymore a diffusion coefficient if you have many particles to arrive to a target, because if you think about the first one will use completely different type of geometry, completely different type of path. And this could lead to very high activation because you have many particles. The first one will arrive is the one that will select this optimal path. Think about fertility. It's an important process, right? When a sperm enters the uterus. Uterus is the place where somewhere you have the egg. You have so many sperms that if those who are deviating from the optimal path, they will miss the target because the one that will follow this optimal path will win. So this is a different, and we think it's a general picture about signaling. Signaling has to do with this fastest particle. Yeah.
But why is this not for your simulation for the presynaptic side? There you have the same.
Absolutely, we have the same. We have the same problem everywhere, because this we did this year, and the other one we did 5 years ago. And now we have to revisit a lot of what we have done. So just to finish with this, if Kanishka will explain that if you have an obstacle, you see that the optimal trajectory avoid the obstacle, and they use the shortest path, the shortest trajectories, and they don't anymore, this like the mean first passage time, visit as a typical trajectory the entire domain. They shoot toward the target through the optimal path. And we think it's a general principle of signaling. Signaling has to do, and that's why you need redundancy. So we have now a review that will hopefully appear. I mean, we have to review it first, and hopefully it will appear. About we discuss extreme statistics in biology, in several examples of biology, to explain how signaling, the time scale of signaling, is given by this initial redundancy to find a small target where you have this amplification. Okay. So I'll stop here. We discuss pre and post synaptic, where presynaptic calcium nanodomains between vesicles can be influenced by a parameter, the density of vesicle, the location of calcium channels. Calcium dynamics add randomness in vesicle release, and according to what we have heard this morning, there is an entire complexity that we haven't touched, that Ben has been worked out about this other stochastic event due to this synaptotagmin. There are different types of synaptotagmin, synaptotagmin 1 and 7, and it is known to be associated to triggered release or spontaneous release. It's also something that could be, would be interesting to merge all of this in the end, to have a general theory about what defines the release probability. And so I just showed you this ER calcium dynamic here, but I don't want to say too much. But just to finish about this extreme statistic, and we think it's a general principle about signaling. Yeah. This is something that I wanted to say, but since I changed the talk, I would say this to finish. Kanishka will show you that simulations and analysis can lead to prediction. And in our field, this is always very hard, because when you have to compete against experimentalists or to explain that maybe you have found something, this is something very hard to convey, that there are predictions. We made several predictions in the past coming from modeling, and we and others. And I think this is very important at the stage of our community to convey this message. We can predict, and we can predict even in advance, and Kanishka will show you some result on that. Here, many years ago, we predicted the column organization where we have the presynaptic terminal, where vesicles are released, that are exactly colocalized where the AMPA receptors are clustered. So in 2011, we explained this, that should contain multiple structures where vesicle fusion spots are opposed to one or several clusters of AMPA receptors. So we proposed this idea. And indeed, it was found and discovered using super resolution at least once, review also last year, and there's a paper also this year. So I think this is an important message, that the modeling and simulation, we are ready. We are ready for this, and it is important. I'll thank everybody, Kanishka, Eddie, and all the people in my group that are working with me very hard every day on all of what I showed you today, and especially to my collaborator, Zev Shust, who passed away this year. Thank you.
I have not a question, but just a comment, which I guess is very important. I mean, this principle of first arrival for signaling is definitely a brilliant idea. I think so. And it is applicable in a case that one is enough, or in a case that this first who has arrived can amplify itself, can like create new signal. If this mechanism doesn't exist, for example, in pre-synaptic terminal, we should prove first that magnification mechanism exists. Then it will be definitely applicable. If not, then one doesn't matter actually. So this is just a little comment.
Sure, sure.
Thank you.
I agree.
I have a question and a comment. Now, the comment is another example of extreme statistics. Was a paper about 10 or 15 years ago by John Rinzel and Hagai Agman-Sneer about in the auditory system. I think it was intraoral time differences.
Okay.
So you can add that to your review
Sure.
if you still have time. The question is, you said that you showed some data quickly that blocking ryanodine receptors eliminated the early peak in the base. But are we going to hear about that in the next
Yes,
talk?
yes,
okay.
yes.
All right, because I didn't see how that had anything to do with the extreme statistics.
It will come.
Okay.
Yeah, and if it's not, please ask the question. We will go into the detail of this.
To come back on Martin's question for this study, did you look at the time or the probability that the ions touch first the ribbon before escaping in far away?
We look at both. So there was a time. First of all,
because
if
there
you're
is
in the
a first
boundary
passage
layer,
time.
if you're in the boundary layer, the time is immediate because it's very fast. It's microsecond. If you're a little bit far away, then indeed you are in the regime where you're already in the bulk. So we first have to calculate where you are. If you're in the boundary layer, then the time is fast, and then you have to calculate the splitting probability. But otherwise, if you're far away, then you just have to calculate this time, because this is a time that which
yeah,
is
but this is like an exponential.
That's right. That's right.
And another question is that instead of splitting like the time for the first particle to reach and the mean first passage time, is there a way to compute like the distribution of the passage time so that you will have access to everything? You will have even access to the first passage time of the last particle, for example.
Yeah, sure. So I showed you the distribution of time. Actually, okay. Narrow escape means the time it takes for one ion to reach a particle, to reach a small hole. The time is mostly, if you're outside the boundary layer, the time is long. If you have many particles, actually we went to this exponential time to actually short time asymptotics, because the one that shoots, you have to go back now to this regime where exponential time is irrelevant. And so that's why we had to calculate this entire distribution, this probability distribution function of the time. And this is where we saw that actually the part of the exponential was not anymore relevant. And what is important is the short time asymptotics. This analysis was done started in the 70s by, there was many Russians involved in this time asymptotics for the diffusion. And it has to do with, and Joe Keller and his team, in the US, where they looked at how you calculate now short time asymptotic using the Ray method, which is WKB approximation. So I didn't discuss this, but yeah, we needed to have this entire distribution, and we found out that only the short time is important. Yeah, that's right. You use the Econal equation, and that's why the solution of the Econal equation is the ray, is a direct ray.
So I have a question, maybe a comment, a follow-up on Eddie's comment. So I think I have read somewhere that vesicles, or maybe it was secretory granules, they contain a lot of calcium and apparently can be released. So,
sorry?
Say secretory granules or the vesicles in synapses. They contain lots of calcium. Maybe someone can confirm that. And that can be released. So maybe we have like a calcium-induced calcium release, which then triggers exocytosis. So I don't know if... Anyone has heard about this?
Well,
maybe.
At least we haven't studied that.
No.
But it could be it could be part of this amplification process.
Exactly.
Rewinding to your what did you call them? Your potential wells, the motion of the calcium channels.
Yeah.
Have you? Did you compare? I was wondering, David, if you compared this to any of the literature on. It's somewhat controversial. You know this business of lipid diffusion and fences.
I mean, there is Kuzumi theory of the 90s, but this is different type of. So first of all,
I mean, in other words,
we had lipids.
Yeah.
So Martin gave us lipids also, lipids that are GFP. So we could follow the lipids and compare the lipids. So the lipids we have. I mean, it's true that I didn't show here, but there is a clear difference between the lipid behavior and the calcium behavior, calcium channel behavior. For example, we could find sometimes that lipids could aggregate at some specific place, but the number of this place was 20 times less than what we have here. So there is a real difference between the lipid motion and the calcium channel motion. I didn't show the data here.
Yeah, I could imagine there would be. I suppose, I mean, for me, the issue would more be, is it the cytoskeleton? You know, I mean, the idea there, as you know, is you've kind of got these fenced-in areas, which would have a different effect, presumably, on every type of species. A lipid is very, very, very different to a to a channel, obviously, and would experience a different kind of fencing in. But it's very controversial whether that's even for lipids, it's highly controversial.
So I think it remains a very interesting question to understand from an experimental point of view. Maybe we should have asked Fred Pense, or how can we address the question about this? What defines this accumulation of receptor? Even if we don't talk about potential well, why you have such restricted domains where you have a high density of these channels that exactly colocalize where the synapse is, the postsynaptic density is, or the vesicle is, and that it can extend to tens of nanometers or hundreds of nanometers. I think you're right, it has to do probably with some actin postsynaptic density scaffolding organization. We started to look many years on this. We didn't succeed to get any conclusion. Probably we need people that are more knowledgeable than us about membrane organization, actin organization, to try to understand how something then can pull and create a force tens or hundreds of nanometers away. And it's not clear to us what's the physics of this.
I mean, maybe I have a comment to this. It could be also that you have several release sites, which is true for all the synapses. You don't have a single release site. You have 5, 6, 8 vesicles that can go out in this active zone. And since we see this morphing of this potential wells within the active zone, it could be that you maybe have a kind of borders set by this predetermined active zones. You have Monk 13 as kind of a place keeper for this active zone, for these release sites, and maybe in between these Monk 13 spots, there's maybe the place where the calcium channel starts to localize or starts to immobilize, and then of course a single one has the chance to go in and out, and the crowd is kind of locked in this place, and because we have this 30-second appearance of these structures and then they reappear on another spot, then maybe they can escape on a certain point out of this ring of release sites, and then maybe you create another release site and fill in the biggest hole, and then they start to morph in this region. So this would be a kind of hypothesis. next speaker will be Kanishka. He will talk about or continue where David stopped, describing this extreme statistics.
Maybe we have to turn some of the lights because otherwise it's too dark. Is it? Okay. Yeah. Okay. So yeah, I think I don't have to introduce that I do a PhD with David, and all the experiments of the talk were done by Eddie. So indeed, I'm going to speak about the spine apparatus and how it especially contributes to these fast calcium transients that David showed you 10 minutes ago. So yeah, first I would start with describing what is a spine apparatus and what are the receptors involved, and how the calcium dynamics are. Can you hear? Okay. Calcium dynamics are regulated by spine apparatus, and finally, most of the things actually David showed, but computational modeling and how I model the spine apparatus computationally, and what I obtained as from simulations, and finally the explanation of using the framework of extreme statistics of calcium diffusion that we are trying to explain this fast calcium transients. So spine apparatus is nothing really new. It was found in the 50s by this paper by Gray, and so here you see the presynaptic terminal and synaptic cleft, and inside the dendritic spine you see this structure. And here in the 50s they wrote like the structure here termed the spine apparatus often appears in the spine cytoplasm, and in the 50s they obviously wrote that no function can be ascribed to the spine apparatus at that point. So then fast forward half a century nearly. So in 2011, what we know about spine apparatus, we know that it's a specialized form of endoplasmic reticulum, smooth ER, and it's present in dendritic spines. We can see with electron microscopes, and still we don't know the role of spine apparatus. So another important thing discovered by this paper by Deller and friends is that there's an important protein indicator about the presence and absence of this spine apparatus, which is synaptoporin. So in this particular paper, in the wild-type mice, so here is the spine, and we can see clearly there's the presence of spine apparatus. But when they knock out this protein synaptoporin and look at the spine apparatus is not present. So we have a clear indication of, we can get a clear indication about the presence or absence of spine apparatus by synaptoporin. And when they knocked out synaptoporin, the mice also showed some deficits in synaptic plasticity. So that implies that spine apparatus is also important physiologically. So then, in order to study this, we did two types of uncaging experiments. So it's always calcium uncaging. So we use either the dendritic head, spine head, or the spine base. So uncaging means you put NPX caged calcium inside the spine and then put a laser and then measure the calcium using flow 4, both in the head and the base. And also it's the same with here. So initially we didn't know whether it was synaptoporin positive or negative, but all the results that I'm going to show from Eddie is with 18 synaptoporin positive synapses and 26 negative synapses. So this is what exactly David showed you. So in synaptoporin positive spines, you see this immediate calcium increase in the base when we uncage in the head, and this delay is about 1 or 2 milliseconds. So this is what we really wanted to explain. And when this spine is synaptoporin negative, and we don't see this increase, so it's almost flat in the base when the uncaging is done in the head. But we also have data from the uncaging in the base, and for this direction it doesn't give any amplification towards the head. So here the red curve is the calcium fluorescence in the base, and in the head you don't see any increase in both synaptoporin negative and positive synapses. So what we wanted to explain was these really fast calcium increases in the bases of dendrites. So for this I made a computational model to study how actually this could work. And the first guess was to do a Brownian simulation, so simulate exactly what was done in the experiments by uncaging 1000 calcium ions here. So here we have the geometry of the dendrite, geometry of the spine that I made. So it has a 1 micron head radius and neck length of 1.5 microns and 150 nanometer radius for the spine neck. So can you hear? Okay.
Just one question about your diffusion coefficient, 600 micrometers squared per second.
Yeah. So that's what we had.
That looks like really, really fast. I mean, usually calcium unbuffered in water is around 200 micrometers squared per second.
Okay, but 200 and 600 doesn't really change. Yeah, for the extreme cases that we looked at. Yeah, usually it's reported 400 to 600. So yeah, the first guess was to do a Brownian simulation and look at how long does it take on average for calcium ions to arrive at the base. So this is the distribution that David showed you. So it answers your question. So this is the whole distribution we fit this Poissonian arrival process. So we fit an exponential for this and obtain the mean first passage time of 120 milliseconds. And it's actually calculated from this formula. Here R is the radius of the spine head, and A is the radius of the spine neck. So, but if you look at the formula, if we decrease A, actually almost all the terms explode. So that means if we shrink the spine opening here, it gets really difficult for the calcium ions to reach here. So that means in this simulation there's actually no spine apparatus, but if we put a block by introducing the spine apparatus, this time will get even slower. So that was the problem that we wanted to solve. So how does it work? So that means the spine apparatus has some kind of amplification going on. So this is the distribution of what is actually present in the spine apparatus. So for calcium uptake, we have the Circa pumps, it has been found, and two types of release channels, ryanodine and IP3. But IP3, it's not really found in spine apparatus. It has been found experimentally, and also Eddy confirmed it, that ryanodine doesn't really, is not part of the game. So in the computational model, I model Circa and ryanodine, so, and model the dynamics of how they really work. So in the left you see the luminal side, and on the right is the cytoplasmic side. So Circa operates by having two calcium ions in this receptor head to open and absorb calcium. So when the first ion arrives, it is bound to the Circa pump, and then it waits for a second one. And then when two ions are bound simultaneously, the calcium is taken into the ER. And ryanodine, on the other hand, is the channel that is releasing calcium. So it also has two binding sites. So when the first one arrives, it waits for the second one, and then it's open and it releases n number of calcium ions. So this n is like the amplification factor of the process, and it's a parameter that actually we varied during the simulations. So then how to model the spine apparatus? I modeled it as a spine inside the spine. So this is actually the geometry, and in blue you see a typical trajectory of calcium ion, a Brownian particle diffusing to the base, and at the boundary we have an absorbing boundary condition, and the number of ryanodine and Circa were chosen as 36, and they are all 10 nanometer absorbing. Circa 10 nanometer absorbing discs, and ryanodine are 10 nanometer calcium sources. So this is the distribution, the placement of this Circa and ryanodine is like the most important thing to discover, because nobody has actually looked at it, and nobody knew it's really important. So the first guess was to think, okay, if you release calcium and if the amplification happens here, ryanodine should be in the base. So if Circa takes calcium into the ER, they should not compete with ryanodine. So this should be as further as possible. So that was like the first guess, and you will see what we found. So then I do the simulation. So the vertical axis is the number of calcium ions arriving at the dendrite. So that means I choose a 300 nanometer box and count how many calcium ions are present in that box at a particular time. So as you can see, to compare with this green curve where we don't have an ER, the red and blue curves with the amplification of ryanodine gives a very strong response, very strong calcium response after 1 or 2 milliseconds. So within 2 milliseconds this amplification starts and then goes up. So what is critical is that 2 calcium ions arriving at the first ryanodine receptor out of the 36, and then it releases n number of calcium, and these calcium released can activate the neighbors. So then it goes to a calcium-induced calcium release. So, and this it is initiated by the first 2 calcium ions, which arrive within 1 or 2 milliseconds. So this is what we found from the simulation, and we looked at the distribution of how long does it take for the first one to open actually. So that means how long does it take for 2 calcium ions to arrive at the same ranitidine receptor. So this is the distribution with when we uncage 1000 calcium ions. So these are all simulations. And then we see a mean time of about 2 milliseconds. But if we uncage less, if we uncage 500 calcium ions, the mean is a bit increased, 3.41 milliseconds. So the opening time distribution of the first one depends on the initial number of ions. So these extremely fast processes depend on how much was initially present, because we have to wait for the first one to arrive. So then indeed we checked what happens if we place Circa also in the base, and it was no surprise that calcium, some of the calcium released by ranitidine actually goes into the Circa bonds. So that's why we see a low amplification when Circa is present also in the head and the base. But if it was only in the head, it will be like this blue curve with the beta amplification. So then we were quite clear about the distribution of Circa pumps, that it shouldn't be in the base. But what about ranitidine? So up to this, I modeled all the ranitidine as calcium sources with calcium sources, and I didn't really model the dynamics of what's going inside. So I treated the spine apparatus as an unlimited supply of calcium, but it's not physiological. So I chose an initial number of calcium, then I let calcium to deplete from the ranitidine and come in from Circa pumps. So if we have 150 calcium ions at the beginning, so this turns out to be about 0.4 millimolar, if I'm correct. And then if we place ranitidine also at the top, you see that compared to the other cases where ranitidine was only in the base, which I did before, it has a very weak response. So the strongest response in the dendrite occurs only when ranitidine is positioned at the base. So what's going on here? So it's actually not really magic, because if we put ranitidine also in the head, the first 2 ions for reaching these ranitidine receptors is very quick. So then it can trigger the calcium induced calcium release already here, and then the stores are depleted really quickly before they actually arrive at the base. So that means ranitidine should only be in the base to get this kind of correct amplification. So those were the predictions that we made, and the remaining thing to do is also to model simulate the calcium uncaging in the base. So when we uncage in the base, the calcium signal should not propagate to this direction. Actually, in the experiments, we saw something really flat. So this is something that the model cannot 100% reproduce, but still, if we see in the case without the ER, there is some number of calcium arriving at the head, about 50 out of the 1000 that we uncaged. But if we place ranitidine in the head, these numbers increase. So that also confirms that ranitidine should only be in the base in order to avoid the back propagation of the calcium signal if we uncage in the base. So then we were quite clear about these two distributions of Circa and ranitidine. And then to summarize, Circa should be in the head of the spine apparatus, and ranitidine should be the base. And then we have this kind of diode effect with amplification only occurring into this direction but not towards this direction. So this is the prediction that we made, and then we did a Skype call with Eddie, so what's going on, to see actually if we can see this experimentally. And this was actually the correct prediction. So I would start from here. So if we compare with the synaptopore in positive synapses with negative synapses, Circa is present quite significantly in synaptopore in positive synapses, and it's the same with ranitidine. And on the other hand, if we look at the location, Circa pumps are located in the head of the spinal apparatus. So in this experiment, Circa and synaptoporine are colocalized here. So you can see with blue that Circa is towards the head of the spinal apparatus. So the synaptoporine or the spinal apparatus is shown in green, and it's also the same here. But on the other hand, ryanodine, it's always at the base. So you see synaptoporine in green and ryanodine in blue. So the prediction is quantified here. So Circa is mostly present in the neck or the head, but ryanodine always in the base. So this confirmed our theoretic computational model's predictions, and then we were quite happy. And the next thing was to really get the timescales of calcium experiments that we were initially produced with. So the first thing to reproduce is what happens if we don't have a spinal apparatus. Then you have a decay of the calcium signal also in the head. So that is basically accounted by the calcium pumps, because it's not just diffusion towards the head, but some are actually extruded in the head itself. So then I calibrated the number. This actually takes into account all the other mechanisms, but so roughly we can reproduce this curve, and the calcium accumulated in the base is almost 1 or 2 calcium ions, so which agrees with what we see from the experiments. And then the next stage is to reproduce the time scale with the presence of spinal apparatus. So if we have about 50 calcium pumps, we can get the decay of the head correctly. But if we look at the increase, so we know what happens here now, because the first 2 calcium ions reach the ryanodine receptors and induces calcium-induced calcium release. But then, how does the ryanodine actually function to get this kind of curves? So there's an increasing phase and then decreasing phase, and we know that ryanodine cannot continuously release calcium because the stores get depleted, and then it becomes extremely hard for the receptor to go against this concentration gradient accumulated by just outside the receptor. So we found that in order to get this kind of amplification, instead of if a rapid amplification like this, we need a delay of about 0.25 milliseconds after the second ion arrives. So that means ryanodine, when it's bound to 2 calcium ions, it doesn't really release immediately, but there's a delay. So that delay should be about 0.25, otherwise you don't get this kind of slow increase. And then indeed the release of ryanodine, nobody actually knows the numbers, how many ions are released by each opening. So in this simulation I started with 8 and 7, 6, and then we kind of see this kind of curve which is similar to the experiments. So we assume that this is we predict that this is like the number of ions usually released by a receptor, and at the end, if we have 36 ryanodine receptors, this turns out to be about like 500 calcium ions at the maximum in the base. So then there should be a refractory period, because if it continuously releases, this response explodes. So the refractory period is about 6 milliseconds after one of these releases. So that's what we predicted, and still we haven't tested this, but the next phase is to look at what happens actually if we have different neck lengths, because in the first equation I showed. Yeah.
Again about your numbers. So the ryanodine receptors that are there, are they significantly different from the one you find in cardiac myocytes?
Maybe I should answer.
I don't really know about cardiac myocytes. Each
I guess it should be much lower here than in heart, because I mean there it's a massive release, while here I guess it's a much weaker one. On the other hand, they may be concentrated on very small area, so I don't exclude that there they should cover big areas. So in this case it can be very focused, but particular numbers I don't know.
A refractive period of 6 milliseconds seems really short. It's almost nothing.
Yeah, but I saw some clamp experiments that they measure the currents. So the delays can be about at this scale of delays. So it releases and then waits for 5-6 milliseconds, and then there's another release. So.
Oh, maybe you're talking about different things about refractive period. To me, a refractive period is you cannot release within that window.
Yeah, so it releases and then stops for.
And then stops. So if you applied another pulse after 5 milliseconds, there would be almost no release. Sorry, after 1 millisecond, there's no release. After 5, you release a bit more. After 6, essentially, it has recovered to its full levels.
So yeah, something. So I mean, here it's 1 or 0. So I shut off the channel for completely 6 milliseconds. But I know it's a curve. It's not like 1 or 0. Yeah,
that's impressive.
Yeah. So the thing that actually slows down all this diffusion is actually the neck length, because so this is a complaint that we got from the reviewers as well. So if you have longer spines, you might not see this kind of statistics in this kind of timescales. But we did simulation with lengths comparable to the neck length, so that we find in spines. So and computed how long does it take for the first 2 to arrive at the end of the spine. So this is 1D simulation, but still we see that the time scale is still at the order of milliseconds, so it doesn't delay that much. And also in the experiments we see that Eddie has data from different neck lengths, so still we see this fast amplification when we do the uncaging in the head. The base always responds really quickly, so the neck length really doesn't modify the millisecond time scale. So that's what we wanted to check. And so to address all these complaints, we have to actually derive a formula to say how does everything depends on the parameters that we used in the model. So this is like the next job to do. So we have these histograms of how long does it take for the first 2 ions to arrive, and then we try to get an analytical formula for these distributions. So I don't go through the whole derivation, but basically what we assume is the calcium arrives at the junction between the spine head and the neck, and then it diffuses in a 1D motion. So we basically convolve the time it takes for this to arrive here, and then this 1D is already known, and this one, this one is a Poisson arrival process. So if we convolve this, we get the probability distribution function for this, and then mean first passage time for the from here to the neck opening is what we should put, and I checked it with the simulations, and what we get from the theory actually quite agrees. So then we can convolve it with the 1D motion, and then for the first one to arrive, once we know this previous function, we just have to do this so 1 minus the Sowell function into n minus 1 lines still in the domain, and then multiply it with the flux. And for the second line, once we know this function f, it's another convolution. So here we assume that time is divided by 0 to t is divided by 0 to s and s to t, and then the first one arrives within s, and the second one arrives within t s to t. But this is not. Then I did the. Then I compared it with the simulations, but it's actually a bit slow because that assumption is not really correct. Because one thing, if there are ions that are reaching here, they can actually go back to the spine head and then come back. So it's not like the straightforward process that I showed you. So we have to correct for that, and the mathematical details are actually in this paper we published recently. So I'm not going to go through all. And then the second correction is when the first one arrives, the second one is almost halfway through, or maybe even closer. So then we have to correct for this, and with this we obtain these black curves that quite agrees with the simulation results. And then we could say that most importantly, how this initial number of calcium ions present in the head regulate the number of time scales. So actually we found a log dependency. So when we increase the number of number n, the number of calcium ions initially uncaged here, the time gets reduced logarithmically. So that's what we found. And as a summary, so the calcium signal from the dendritic head is amplified by ryanodine receptors through a calcium-induced calcium release, and their receptors are located in the base of the dendrite, base of the spine apparatus. And the first ryanodine receptor is opened by the first two calcium ions arriving first, and these numbers, these arrival times are modulated by the number of calcium ions we uncage initially. So if we uncage more, we get faster time scales. So then the second part of the talk is about the trajectories. And so in all our analysis, we didn't really take into account the presence of spine apparatus because it was just diffusing here and then going through a straight line. So what happens when we have obstacles with these extreme statistics? So that's what we wanted to study. And for this, I went back to a very theoretical simulation with a very simple domain in 2D. So we have a release site here, the source, and then a target. And in between, we have a circular obstacle which is reflecting. So we wanted to see how the fastest trajectories look like. Do they really explore the whole space, or is there a way that the trajectory is following when we have an obstacle? And so once we did all the previous analysis, it was quite expected for us, because when we increase the number n, the number initially present in the source, the first one actually arrives really fast. With increasing n, the trajectory is short and the time is also short. So this is what David also showed. So if we have n equals 1000 or 500, first thing is that the mean is decreasing. The first one arrives fast, and then the variance is also decreasing. So that means if you want to send a signal, having as much as particles would increase also the reliability of the time. So these are how we looked at these in with a larger number of particles, and at the end is, so this is the log plot. So we have a log dependency of the time, and the trajectory is also decreasing. At the beginning it's really fast, and then still it decreases at least in these kind of domains. So then we looked at how these trajectories actually deviate from the optimal trajectory. So optimal trajectory for this one would be to follow this tangent and then follow the this curve and then come straight line. And then if it actually followed the trajectory within this given number of time steps, it should have gone like this. Then I compute the L2 norm of the distance between the optimal trajectory and where it actually went. And then I also found that this distance, this average distance also gets low and low. So that means the first particle actually follows the optimal path when we have more and more at the beginning. So the last thing is the dynamics of the particle arriving at the target. So then I was wondering whether if we have a target and we release from here, the statistics might be different. So it might not be coming from the distribution, but the particle might be jumping from 1 or 2 steps towards the target. So there are 2 ways to go to the target really fast. Either you come from the end of the Brownian distribution, so that means you take longer time steps. The other one is to follow the optimal path. So in this kind of regime, we found that on average the Brownian simulations have a mean of 35 and variance of this amount of the path is on average one time step is like this, and even if we look at the fastest ones, they are not very different. So that means they still follow the similar, they still come from the similar Brownian normal distribution, but they go faster because they follow the optimal path. So yeah.
But the amplitude of the jumps are, does it depend on your time step also, because this is like a discretized Brownian motion, so the amplitudes of jump are...
Yeah, but on average it doesn't really jump, so that's what I mean, it's not like, it's always moving fast. So sometimes it moves fast, but sometimes it moves slow. So the distribution is not very different from an average particle diffusing inside. So I mean, these are from the first ones. So first ones are not very different from the average one. It only follows the optimal path. So.
There's a basic point which I'm not getting here. So when can you go back to the where we were before? So the statistics of the first particles. Can you obtain those from solving the diffusion equation with the appropriate boundary conditions? Or is it beyond that, and you have to account for the discreteness?
We didn't do the case with the obstacles, and also for this time we didn't actually solve it. But
it's a different question. It's a matter of principle. You know, if you were able to solve this, solve the diffusion equation with the appropriate absorbing and reflecting boundary conditions on the appropriate surface, can you get these fastest particle times? Is it contained in that formalism?
Yeah, so that's
or do you have to discretize it? Is it something to do with you have to respect the non-continuum nature of the field? It's not. So it's actually contained in the diffusion equation with boundary conditions.
Yeah.
I see. Okay.
So in these formulas we didn't assume anything about discretization. So this is without the obstacle, but still coming from the spine to the base. So
I thought you were actually, well then I misunderstood these simulations. So here you're just solving, I don't mean just to trivialize him, but you're solving the diffusion equation with the appropriate boundary conditions. That is what you're doing.
Yeah, exactly.
Okay. Yeah. But if you're doing that, how do you have a finite number of... you know, if I solve the diffusion equation in bulk, I have from a delta function initial condition, I have a Gaussian spreading outwards, right?
exactly.
So how do I decide that one particle has arrived somewhere? I have to say I have so many particles there, and then there's a certain amplitude threshold defines the arrival of one particle. Is that kind of what you're doing?
Yeah, so one particle arriving at the target means the remaining particles are still in the, still not arrived. So that's why we have this survival function times to the power of n minus 1. So this is how we really compute that the rest didn't arrive. So that's the probability of one particle arriving at the target.
Yeah, just to comment on this question. So here what we found is that, first of all, the classical diffusion formalism fails.
So this is my question.
Yeah, it fails. So in order to analyze this, you have to go to pass integral and Wiener representation, where you do a delta t analysis, where you look at the steps. And then there is another steps.
But if I can interrupt you there for a second. I mean, that you know, the path integral formalism is just another way of writing the, you know, it's the same thing, right? So there must be something else, yeah.
So I just wanted to continue on this. So we looked at, because if you want to select one of the first trajectory to arrive, how do you know that it's the first one? You have to look at the trajectories, the entire history. And then what we found is variational principle. That is, it's different from the classical large deviation. It's not contained in the classical large deviation, where, for example, you have a small parameter like in the escape, activation escape, where the noise is small. And if you look at the Wenzel-Fridolin functional, you have exponential of 1 over epsilon, and there is an energy, which is the integral with the pass, the Brownian or the Wiener pass. So we found here that actually the number, the small or the large parameter was the number of particles, and from this you find out that there is a term in the exponential, and you select. There is a minimization principle. You have to minimize the pass. So if there is an obstacle, then you take the geodesic, and so that's why Kanishka showed the picture where we wanted to look as. So now we varied, you look at different delta t, and you look at the shortest one, and the shortest trajectory is actually following, that is how you go from one point to another, you converge, and I don't specify here the mode of convergence because it's not clear to us, but you converge towards the geodesic, and all of this deviation goes to 0. And so what you get ultimately in the end is if you go back to what you showed as a function of time, you know the geodesic is usually you go to constant speed. And so that's why if you go to where we were, we looked at what is the distance along the path, and the distance along the path converges toward this type of constant speed. So suggesting that, again, it's no proof here. There's no proof. We just found this variational or minimization type approach through this non-conventional path integral method.
So just to clarify, so you had a variational principle, you looked at the minimum, that gave you the optimal path, but then the trajectories that you showed on the previous slide or the slide? Mm-hmm. No, there is now. Yeah, hold on. Those are results from direct numerical simulations.
Yeah.
And so you show us that they actually are close to the minimum path obtained from the variational principle.
Yeah, okay. So minimum path would be something like this.
Yes.
Yeah.
But if you, let's take a simpler problem than this. You know, the simplest problem I can imagine, you know, with the first passage problem is just one-dimensional diffusion with a brick wall, absorbing boundary conditions. So the same issues would come up, right? You can ask the same questions. You don't have to have this very complex geometry.
But in one dimension, okay.
I'm just taking the simplest thing I can possibly think of, which you can really test with relative ease. Yeah. So I've got a one-dimensional geometry. absorbing boundary condition, I know how to solve that normally, right? You know, I just write down the diffusion equation. I have P equals 0 there, and I can do everything. So let's say I have n particles, and I ask for how long do I have to wait for? I'm sorry, you know, maybe this is a bit of a tough question. You're standing on your feet, but you know, I have n particles. How long do I have to wait for the fraction that have been absorbed to reach 1 over n? Right? That would be the naive continuum approach, and that's when one particle has been absorbed. So does that give the wrong answer? That's my question. Do I have to retreat from the continuum approaches? I understand that I can re-express the diffusion equation with absorbing boundary conditions in a path integral formulation. You can do that, just like you do in quantum mechanics, right?
Yeah. For 1 over n, we cannot really get with this formalism, because we have to wait. We assume that the first n minus 1 are not arrived, so we have to have the number. So it's not the n is not.
No. What I said is covered by the absorbing boundary condition. It's a first passage problem. So in the continuum framework, there will be, you know, when the total current integrated over time at the absorbing sink equals 1 over n, that means
n means the number of particles, right?
N is the number of particles. Then that means the fraction of the normalization which has gone into the sink corresponds to 1 particle. That would be the naive continuum way of assessing when 1 particle has arrived. I'm just wondering if that's wrong or gives a different answer to this. I mean,
so to solve that would be to do the convolution here, n minus 1 over n times.
I don't.
So if you have two.
That's why I was thinking of a very simple geometry, not.
Yeah. So in 1D geometry, I showed previously that you can have the analytical solution, and you find in the solution that it goes through the ray, and the ray is exactly the path, the optimal path.
But does it correspond to what I just said or not?
But
that's my question.
If here they are all independent particles, so you just look at the first one to arrive. That's it. And the first one to arrive, use this geodesic.
Yeah, okay. That's good. Okay.
Yeah, so this is like the last summary of the second part. So what I showed was tau 1, the first one to arrive, the time for the first one to arrive, and its variance decreases with increased n. So when you have more and more n, the time and the variance decreases. And for large n, the trajectories converge to the optimal path. And finally, so this is the important thing for biology, that the fastest particles diffusing to a target does not really explore the whole space, but instead it takes the optimal path towards the target. So that's the summary from the second part.
Thank you. A further question?
And I suppose that's more a question for Eddie than actually for you. So you showed that there is expression data that shows that the circle pumps are more located to essentially to the spine head, and that the radiation receptors sit at the base. Can you actually monitor ER calcium to see that there is a dip in ER calcium, that you are really sure that the calcium comes out of the ER and is not an artifact, or doesn't come from anywhere else? So where you see this nice, essentially for IP30 receptors, you see this scrap in the ER.
It's a very good question. I wish I'd say that I can distinguish such little, calcium fluctuations. In some cases, I mean, I'm simply, afraid to be a bit bias in this case. You know, in biology, it's very easy to push data to the direction you want. And at the moment you get some theory, some idea, then, I mean, you think you are still unbiased and objective, but it somewhat affects your mind, you know. Actually, it was a big issue for me to make it in most objective way. So I gave the data to our new student who is completely naive and he doesn't know what he measures. And this is the only way to avoid all possible mistakes. And surprisingly, you know, it came out more or less true that if you simply use a caged calcium model, which is very good, because, you know, you don't involve any other mechanisms. Because if you do, for example, glutamate uncaging or whatever, it's very hard to give some clear interpretation. Also in calcium uncaging, it's not so easy because you always have to remember about calcium-induced calcium release and circopumps working there. What is helpful there is that in many cases we don't mark the spine apparatus, which is, you know, first of all for technical reasons, because it's green typically, and also calcium dye is also green. So we try not to put them together. So we do very hard and complicated experiment instead. So we do it in blind. So we don't know which spine is positive and which is negative. Then we record whatever it is and fix the cell, make immunostaining for this synaptopodin, and only after that we decide whether spine was ER positive or ER negative. So it looks correct in general. After this, with many cases which don't fit, but still general tendency is to see truly release in a case of ER there and release at the base. And best proof actually for it is very simple experiment when we block stores. So you can take same spine, not the other one, but exactly the same one, and apply either a high concentration of ryanodine, which blocks this calcium-induced calcium at the release, or also tapsigargin, which slowly depletes stores. And in this case, we can compare exactly same guy, before and after, and we see quite clear contrast between these two cases. And if it is applied on ER negative spine, then we don't see any contrast. So it looks to me, I mean, I always try to say many buts or many perhaps or most possibly, but in this case it looks true. You know, that's what I think. For me, actually, David's idea was also surprising because I didn't think about such a mechanism. I was only recording whatever it is. So it was very nice to find this interesting mechanism, which was not expected. You know, we didn't think about it. So this is an excellent case when theory helps experiment and makes some predictions, not only simulations. So that's why I like it very much, because of this prediction part. Yeah, that's all. Thank you.
And we will come back to the synapse. And
okay, yeah, so I was just in communication with some people at UCL trying to, was Eula actually, and they mentioned that there was this meeting, so it was fortunate that I was told about it. So I got an invitation to come a few weeks ago, and yeah, this was great because I've done different types of modeling, from Hodgkin-Huxley modeling to synaptic modeling, various aspects of it, and it's been good to see how people approach this problem. So I work in Angus Silver's lab at UCL in the Neuroscience, Physiology, Pharmacology department. Let's see. So a brief overview of what I'm going to talk about. So I'm going to give a brief overview of the software that's been developed in Angus's lab called D3D. So it's a reaction diffusion simulator in 3D space. I'm going to then go on to talk about the latest development of D3D, which has been Monte Carlo simulations. So these are hard sphere, non-overlapping Monte Carlo simulations, which we use to study vesicle mobility in the mossy fiber terminal. We looked at the diffusion coefficient of the vesicles and also the vesicle supply rate to the active zone. So those are the two examples I'm going to talk about. So a brief overview of D3D development. So the people involved are Angus, obviously, and Tom Nielsen was Angus's PhD student who wrote the first code for D3D for his PhD thesis. David DiGregorio been heavily involved in development of D3D, and some of his code actually was from his PhD, looking at the neuromuscular junction. Yep.
There's also software D2D. Is this somehow related to this, or this is completely?
It's different, yeah. Yeah. And Zoltan Nuzler, who did a sabbatical in our lab, who started the vesicle mobility project. His student, Latsi, was involved in starting the Monte Carlo simulations. Right, so here's some background about the synapse that we often study in Angus's lab, the mossy fiber to granule cell synapse, which is in cerebellum. So we have mossy fibers coming into the cerebellar granule cell layer and making contacts with the granule cells. The granule cells have about 4 dendrites that extend out, and then they have these smaller claws that reach out at the end. And these are actually within a glomerular structure here, which is ensheathed by glia. And so here you have the mossy fiber terminal coming in. It's quite large, it's 10 micrometers in diameter. About 50 granule cells make contacts within here, and each granule cell contact has about 5 contacts, so 5 postsynaptic densities. So in total, there are about 200 to 400 active zones in the mossy fiber terminal. So there's 2.5 active zones per micrometer squared. It's been measured. So the first model in Angus's lab was a model of the glomerulus. So for this, we had Tom Nielsen did this model. So we have a complicated structure. We have lots of dendrites, which are these cubes here, with active zones presynaptically and then postsynaptic densities. So the geometry was constrained to give the appropriate density of postsynaptic densities. And with the simulation, we have glutamate sources, glutamate detectors, which are the postsynaptic densities, and this is a finite difference simulation. So very basic, but the driving factor, I guess, for creating this this model was a complicated structure. So with this model, they were able to show that synaptic vesicle release from neighboring synapses or active zones can cause what they call a spillover current. So here is a direct current, and then here is a spillover current. And they were also able to estimate the diffusion constant of glutamate in the synaptic cleft. So that was the first model. And then when I joined the lab, I joined David in this project looking at AMPA receptor desensitization. This is a glutamate encaging project. So we had a large laser point spread function, which we would shine over an entire granule cell dendritic head, which is the width of this point spread function was about 9 micrometers. And we would encage a large area of glutamate. And what we found is that when recording from the granule cell, we see the AMPA receptor current would quickly desensitize, but then there was this resistant component to desensitization, about 17%. Right, so with this, we wanted to show that when we're uncaging, that we have what we hope is a step change in glutamate surrounding the terminal. So we did the simulation, we did a large geometry, and then we added illumination point spread function, and we added photolysis and glutamate detectors, and this was also done in finite difference. And with that, we showed that we had saturating levels of glutamate around the mossy fiber terminal. And then, so that was part of that project. And then, same project, we had a small spot, so a smaller point spread function, which we then tried to stimulate single active zone sites. And using simulations, we showed that the resulting currents, the photolysis EPSCs, were approximately the size of the mini EPSCs. Here's another project that was done with D3D. So this is out of David's lab, and some of work started earlier with Angus, looking at the calcium channel clusters. So here in D3D, we added sources, we added uptake, we have buffers and detectors, and created these simulations, and these kind of simulations have already been discussed, but this geometry is rather simple. But with this, they were able to come up with this perimeter release model. And then recently, we were looking at vesicle mobility in the mossy fiber terminal. We used FRAP, and in this case, the vesicles are 50 nanometers in diameter, so they're relatively large. And we wanted to simulate the Brownian motion, so we added Monte Carlo hard sphere simulations to D3D. Again, we have photolysis with illumination point spread function and fluorescence detection. And then we wanted to simulate vesicle supply to active zones. And so in these simulations, we also added tethering, docking, priming, and release to see their effect on the vesicle supply rate. Just to say that, so the D3D has not been officially released, There's been ongoing debate whether we release it or whether we write a paper first. But with the eLife paper, we wanted to submit the actual code that generated the simulations. So we have that, and you can download it. And you can actually use the code now with a few adjustments. But we're hoping to have D3D out released on GitHub. Here's some features of D3D. It's written in Java, so it's easy to write and compile the code. It's object-oriented and platform-independent. Comes with a GUI. It has log files, so anytime you change a parameter, those are logged, and you have simulation runtime parameters you can print out. You can create init files for your geometries and for your projects. And this is how basically people in David's lab and our lab have used D3D, is to create these init files. And that really helps with reproducibility. Input and output, so you can save points with functions, geometries, and projects. And then there's log files. There's a parameter file that's saved with each simulation. You can run batches. So if you wanted to vary one parameter during simulation, you can run it without the GUI on a cluster. So if you have thousands of simulations, you can do it that way too, which is what I did with the Monte Carlo simulator. So again, just arbitrary geometries, photolysis features, and now the hard sphere Monte Carlo. The diffusion algorithms are the finite difference, Monte Carlo hard sphere, and so next we're hoping to do a hybrid where we can simulate vesicle collisions while running finite difference, calcium in the background. So here's a screenshot of the 2D viewer of Tom Nielsen's geometry. This is looking at, this is in the synaptic cleft, looking at the postsynaptic densities, the green, with a single vesicle release site. And you can change views. So the 2D viewer is like going through different sections. We added the 3D viewer at the beginning, but it was too complicated because of memory problems, and you have to reduce all the voxels to be able to see things. So we've gotten used to visualizing things in 2D, but in future it would be nice to have a 3D viewer. Also on this 2D viewer, you can run the simulation and preview it if it's small enough, and therefore you can create movies with this. And there's also a parameter tab where you can go through all your diffusants and sources, your geometries, and you can look at every parameter in the simulation. And the ones that you can change are in white, so you can enter values here. And then when you hit run simulation, all these parameters are saved to a file. So here are the output files here. We have detector output files and the log files. And here's an example of the configuration files with all the parameters, which also comes in XML format. And then the output files can be text files or binary files, which can be imported to any MATLAB or Igor for visualization. So the basic structure of D3D is 4 packages: a core, where all the core code is, the GUI code, the init files, where user init files are stored, and then some utilities. The important class would be the master class, which controls the GUIs, the projects, everything, running the simulations, air handling. And the master comes, the functions are pretty easy to use, so you can say you want to add a diffusant, you can add a source, add a detector. So the idea is that users could go and just pretty much create their project using master. So here's an example of a user init file for a basic geometry. So you would set the project simulation time, the voxel size, You usually have to create geometry, but if it's just a cube, that's automatic, or a sphere. But this was a complicated geometry, so created just a function. But the geometry is pretty easy to configure. It's just ones and zeros in an array, a 3D array. We have coordinate objects, so X, Y, Z coordinates that control where the sources, sinks, detectors are placed. And that's what's done here. And then that's it. So the init files can be fairly easy, and these can be copied. So there are already some basic init files that come in D3D. But there are many reaction diffusion simulators out there, and don't know where D3D fits in with this. But we like D3D. We're going to continue to develop it, and maybe when we release it on GitHub, we'll get more people involved. So future development will be the hybrid 3D viewer, and we have collaborations ongoing. So here's the first use example. So measuring vesicle mobility via FRAP. So we have these mice that have fluorescence protein attached to a VGLUT1 protein, VGLUT1. So synaptic vesicle will have about 9 of these fluorescent vesicles on them. And so here's the brain of a VGLUT1 knock-in mouse. So we looked at the cerebellum, took slices of the cerebellum, and we performed FRAP. So our illumination point split function was fairly small compared to a large mossy fiber terminal. So this is our bleaching protocol. We have 3 initial bleaching pulses, small ones that measure baseline fluorescence. Then we have a large bleaching pulse, very brief, and then followed by small bleaching pulses to measure the recovery of fluorescence. So we make one measurement, and then we make another measurement, and we take multiple measurements in different locations at different Z depths. So here's the recordings from one mossy fiber terminal. The single recordings are in gray, and then the average is shown in black. If you omit the bleaching pulse, that's in red, you can see that our probe pulses that monitor fluorescence are not creating a large amount of bleaching. So with FRAP, the typical approach then, if you have your fluorescence recovery curve, is to fit that with an analytical solution to the diffusion equation from Axelrod. And this equation's here. So we fit that. So the control data is in black. So what I didn't tell you was that there's a small amount of tissue drift in the slices. We measured that and did a correction of the recovery curve. And so we fit Axelrod to both conditions, and they're not dramatically different, but the fit to the drift corrected curve gave 0.025 micrometers squared per second. The recovery came to about 67%. So motivations for the D3D simulations, we wanted to verify that Axelrod's analytical solution for D-long was correct. There's several assumptions based on that solution, including what the point spread functions are. And then we wanted to investigate the impact of organelle crowding on vesicle mobility and mossy fiber terminals. This is mostly the steric and hydrodynamic interactions. So in D3D, we have very simple bleaching reaction. And we have based on, well, the bleaching reaction is based on the illumination point spread function. So our illumination point spread function was measured using a bead. And as you can see, we have the spherical aberrations. The confocal point spread function is just, can be basically described by a Gaussian function. So the bleaching is a function of the bleaching rate and also the illumination point spread function. The fluorescence detection is a function of fluorescence of the vesicles and the confocal point spread function. Okay, so the red curve is the fit to Axelrod's equation. And then with our D3D simulations, we did finite difference simulations using our point spread functions. And we did a search for the best D long, and we found that that answer was pretty close to Axelrod. Right, so now we wanted to sort of tease apart the effects of steric interactions. So in order to do that, we did Monte Carlo simulations. So we needed to know the vesicle density, the mitochondria density, and the vesicle diameter. So with EMs, we computed that the vesicle diameter was 49 nanometers. The vesicle density was 17% in non-mitochondrial space. And the mitochondria occupied 28% of the volume. And with those parameters, we created D3D. So this is just a slice through the simulation. This is the point spread function in the middle. These are mitochondria. And we have vesicles in green. Those are the mobile ones. And then we have immobile vesicles in the gray. Here's our data, and then here's our Monte Carlo simulation, repeated the same about 10 times. Right, so we have two unknowns that we wanted to know. It was what is the vesicle diffusion constant in the absence of collisions? We call that D short. And we wanted to know the immobile vesicle fraction. So we know from the Frapp recovery curves it's about 30%, but it's not exactly, this doesn't tell you exactly immobile fraction because the immobile fraction affects the diffusion constant, D long. So it's going to be near there, but not quite. So with that, we did a bunch of Monte Carlo simulations where we varied the diffusion constant, D short, versus the immobile fraction, and found the best match, which is shown here. So the D short was measured at 0.06 micrometer squared per second and 25% immobile fraction. So using those values, we ran the Monte Carlo simulation and looked at the mean square distance to compute the diffusion constant as a function of time, which is shown here. So at early times, the diffusion constant of the vesicles looks like D short, 0.06, but then as time goes on, the vesicles collide with each other. Their effective diffusion constant slows down, so we call effective diffusion constant D long, and that approaches 0.025 micrometer squared per second, which is what was measured with Axelrod. That's shown here, so we get a close overlap with the Monte Carlo simulations, where D short is 0.06 micrometer squared per second, and the finite difference simulation here closely overlaps, which also matches Axelrod. So what does that diffusion constant look like compared to other cells? So it looks pretty high. So these are ribbon synapses in the retina, which are known to have fast vesicle mobility. So the mossy fiber terminal is comparable to that mobility, whereas neuromuscular junction is significantly slower, as well as estimates of vesicle mobility in the small boutons. But this is, these estimates are quite spread out, shown in pink, because it's difficult to measure vesicle mobility in small terminals, so you can't use FRAP. But there's one study here shown in this triangle here, which did single tracking vesicle and reported this mobility here. Right, so with the Monte Carlo simulator, we ran a bunch of simulations. We were thinking, well, this is quite a useful tool, so if other people have different synapses with different densities, with different immobile fractions, it would be nice if they could just come in here and sort of pick out their density, pick out their immobile fraction, and find out what kind of steric interactions they expect to see. So we ran a bunch of these simulations. We also verified here that our Monte Carlo algorithm matches the original code, Monte Carlo code from Sokoki. We computed this curve also with mitochondria. So this point here is what mossy fiber terminals, the density, and this ratio here, d long over d short. It's the ratio. So next, we were curious about hydrodynamic interactions. So this d short is the diffusion constant of vesicles in the absence of steric interactions, but that includes interactions with the cytoplasm and hydrodynamic interactions as well from neighboring vesicles. So we went to the literature, the hydrodynamic literature, and we found some equations for showing the vesicle density, showing the hydrodynamic effects, and this is how they refer to it. It's called d short over well, we're calling it d short over d cyto, but they don't look at it in cytoplasm, but probably water. So with this, the red curve shows this combination of experimental and theoretical work from this paper here, Takayama. This is for 0% immobile vesicles, so just vesicles moving around. And then we incorporated what happens when you have immobile vesicles using this paper, Fried, who looked at the hydrodynamic effects of having fixed spheres and having a vesicle move through that boundary. So with that, with this equation here, we were able to estimate the effects of hydrodynamic interactions given our vesicle density in the mossy fiber terminal, which is about 0.5. With those two numbers, ratios, d long over d short and d short over d cyto, we can derive the relation of d long over d cyto, which is shown in this paper here. It's a simple multiplication. So that's the multiplication of the two lines in the previous figure shown here. And when you do that, the total effect of hydrodynamics and steric interactions is about 0.2. So with that, we did some calculations. When we computed, since we know d long is 0.025, we estimated that the diffusion constant of a single vesicle in cytoplasm is 0.127 micrometer squared per second. So how does that compare in water. So this is computed via the Stokes-Einstein equation, 12.7. And then there was a study that where they injected beads into the cytoplasm of 323 cells and looked at their diffusion constant. So these are dilute. And so because they're dilute, then you can say that those are approximately d-cyto, and those were about 0.45 micrometers squared per second. So our estimate is about a factor of 3 off, but since these are beads, then we don't expect them to have the same. Right, so in our paper we gave a table. So we wanted, based on these results, these two figures, we wanted to see if we could estimate the long-term diffusion constant in different terminals. So we did some number crunching. Neuromuscular junction, we found the densities, we did some calculations, and we estimated the long-time diffusion constant, 0.006 micrometers squared per second, which matched closely what was measured. We did that with the boutons. So in the boutons, the vesicles are usually clustered around the active zone. So we found the density in the clusters and computed the diffusion constant, which is also very low. We did a ribbon synapse. Now, we didn't have all the complete information, but in this one terminal, we estimated it at 0.014 micrometers squared per second, but that and measured in that one terminal was 0.11, so that is off. But in the other ribbon synapse, it was closer to that value. Right, so in summary, 75% of vesicles within the cerebellum ossifiber terminals are highly mobile, with mobility comparable to that measured in ribbon synapses. Vesicle collisions and hydrodynamic interactions arising from organelle crowding are major determinants of vesicle mobility. So in the next example, I'm going to be talking about how we used D3D to simulate vesicle supply to active zones using the EM reconstructions that we had. So the motivations for these simulations, we wanted to know, is vesicle diffusion to the active zone, is that process rate limiting during high frequency stimulation? And then how much do steric and hydrodynamic interactions affect vesicle supply to active zone, out of curiosity? So some background, a study out of Angus's lab, Chiara Savian, did these recordings from mossy fiber granule cell synapses where she, at 100 hertz, stimulated them for a very long time. And using that, she was able to create this model of the different pools and reloading rates to the active zone, which is shown here. So the model is that there's a large reserve pool sitting in the back. For a single active zone, there are about 500 vesicles, but within the mossy fiber terminal, there are thousands, so it's quite large. But this is for a single active zone. Then you also have a releasable pool, which is nearer to the active zone, and that's large, it's 300 vesicles. And that was computed via Chiara's data. And then the reloading rate from this pool to the active zone was computed at 40 to 80 vesicles per second. This reloading rate includes the time for vesicles to move to the active zone, presumably via diffusion, docking, and priming. Now, the ready releasable pool in these terminals is quite small. The active zones are small. So the ready releasable pool is 1 or 2 vesicles. So Zoltan did some reconstructions of the active zones. So there were 14 reconstructions. So we have the outline of the active zones as well as the vesicle positions. The density of the vesicles is next to the active zones is higher, and then it falls down and approaches that of the density that was measured at the middle of the terminal. So using these reconstructions, I imported the active zone locations and the vesicle locations into D3D. Now there was, as you can see here, there are invaginations. So there, with the EM reconstructions, there's space, like you can see here, around the vesicles, which, if you include them into the vesicle density computation, you'll find that the vesicle density drops off quite quickly. So with these simulations, we assumed that that was what we thought was non-diffusible space, because there were no vesicles there. So the idea was that the vesicles seemed to be clustered and what we called a cloud around the active zones. So basically what we did is we collapsed the space that didn't show vesicles around the vesicles. So we did that in D3D. So the diffusible space was restricted to within the vesicle cloud. And then we added a reserve pool of vesicles surrounding this. So at the very ends here, we allowed vesicles to come in from the reserve pool. We made 25% of the vesicles immobile as we computed. And then we let the vesicles move in Brownian motion. We gave them a diffusion constant, d-Cyto 0.127 micrometers squared per second. Now, so what we did then in the simulations is that once the vesicle contacted the active zone, we immediately released the vesicle. So in this way, we are measuring directly the vesicle supply rate. Now, because the vesicle density around the active zone depletes, that means that the vesicle concentration changes, which means the hydrodynamic interactions changes. So we created an algorithm that computes the local density around each vesicle, that then computes what the hydrodynamic interaction is and adjusts d short. And we also included hydrodynamic interactions effects from the active zone wall, so that as it approaches the active zone, the vesicles slow down. And then we also had a couple other configurations for these simulations. We included vesicle tethers. So if the vesicles were within 150 nanometers from the active zone, we tethered them together. From the distribution that was measured from a paper, 1.5 connectors. On average, there were 1.5 connectors per vesicle. The tethers were 10 nanometers in length, and they had the on and off rates shown here. And then we also tethered them to the active zone, shown here in blue, and we allowed 2 tethers at a time. And then finally, we wanted to know what the effect of having this closed geometry around the vesicle clouds. So we removed that and did the simulations as well. Okay, so this is under control conditions for the 14 active zones. So that each individual active zone is shown in gray. We're measuring the vesicle supply rate at the start of the simulation as a function of time, and we ran the time as long as Chiara-Savian's experiments. And what you find is you have a very fast vesicle supply rate at the beginning, and then it drops off and falls to about 8 vesicles per second on average. Okay, here we looked at the same simulations under different conditions with connectors. So with connectors, it initially slowed down the vesicle supply, but then in long terms, it did not have an effect. We opened up the geometry. So in that condition, we had lots of vesicle supply, and then that tapered off at another high supply rate. And then we also looked at the effect of hydrodynamic interactions and steric interactions, and when you remove those, no surprise, but you get an increase in the vesicle supply, and the effects of steric interactions are not seen until later, until after a few seconds. So with this data, we also estimated this should be the releasable pool, not the RRP. Using back extrapolation, you can estimate this is what's typically done for experiments where you have these cumulative release functions. You can back extrapolate to time 0 to estimate your releasable pool. And when you do that with the control case, you come out to about 200. So here's a summary of comparisons, and we compare it to the results that Chiara had. So at early times, on average, the supply by diffusion is very high. At intermediate times, about 100 milliseconds, the supply rate is about 50 to 100 per second, and that matches Chiara's reloading rate. At late times, so at these times, we're looking at supply from the releasable pool. But as time goes on, that releasable pool is depleted, and then at the longest times, you're looking at the supply from the reserve pool, and that supply comes out to be 8 per second, which matches very closely Chiara's data. And then we also gave the pool size here, estimated. So Chiara's data is shown here in gray, and then our control condition matches up quite nicely with Chiara's data. Interesting, when you open up the geometry, so you don't have any, you just have all this diffusional space around the active zone, you get quite dramatically different results, and you don't have a releasable pool. So finally, we wanted to repeat these simulations with docking, priming, stochastic release, a bit more realistic. So we simulate, we have docking, and we have a release probability 0.5, which matches the Mossy fiber terminal. We have 100 Hertz stem trains, and we set the time of docking and priming to match Chiara's data. So it's it's here, so 16.7 milliseconds delay. So that's the limiting case of that Chiara measured. It's that reloading rate. And in these simulations, and then we also plot here the cases where we just have instantaneous release, and those are shown in dotted line. So in this case, at early times, we see that docking and priming are the rate limiting conditions. And then as time goes on, down here, then we see that diffusion becomes the rate limiting process. And that's summarized here. So at long times, with the docking and priming simulations, we still get the same release rate, 8 per second, and we're still seeing the similar pool size. Just confirming that the way we measured the supply rate was accurate. So in summary, at early times during high-frequency presynaptic activity, diffusion-mediated vesicle supply to the active zone is substantially faster than the experimentally measured vesicle reloading rates. Docking and priming are the rate-limiting factors. At later times, however, during prolonged synaptic activity, after the RP is depleted, the rate of vesicle release becomes limited by diffusion. And our active zone simulations predict the size of the experimental measured RP and the vesicle replenishment rate from the reserve pool. And yeah, these are the people that I collaborated with, and yeah, that's it.
A question. What is happening if you change the phosphorylation state of synapsine, that you can kind of release more vesicles out of the reserve pool? Is then the supply rate different?
What about synapsin?
If you just phosphorylate synapsin stronger, that you have more release out of the reserve pool.
Right. So we did some experiments where we measured. We measured the vesicle mobility using oxalic acid, which is a phosphatase inhibitor. So presumably that is breaking up the synapse and holding it, and we got much higher vesicle mobility rates. So there's something that is clamping down vesicle mobility, and it very well could be synapsin. So in that case, we would, in our simulations, we would dramatically increase the mobility of the vesicles, which would then impact on our estimate of the reserve pool. Yeah.
So where do you see D3D strategically? So where do you want to go with it? Do you want to develop it in some sort of one-stop solution for all problems related about vesicle release? Do you want to target it to certain morphology? So what's your vision for D3D?
Yeah, so originally, I mean, the D3D was started a while ago, and so originally, all the other simulators were just earlier in development. So originally, Angus' idea was that D3D could be useful for people who wanted to use it to study photolysis. So incorporating point spread functions and looking at that kind of reaction, uncaging, and FRAP. But now there's other simulators where you can do that. So yeah, it is a question, what's the usefulness of it? I mean, I think it's easy to use. So I think for people using finite difference just for the calcium simulation is quite useful. If you just want to do the simple calcium sources and detectors, and then creating your own geometries is quite easy. So, but I guess with the Monte Carlo, my vision would be to do the simulations of calcium finite difference along with the vesicle Monte Carlo.
Yeah, it was just a general question about the active zone. Active zones.
Mm-hmm.
I wonder if you have a sense of what's going on with the cortical cytoskeleton. I mean, presumably where there is an active zone, there's a clearing. There's a hole in some sense in the actin cortex, which sits, adjacent to the membrane, to allow, exocytosis. Have you, visualized anything that illuminates that?
With our EMs?
Yeah.
No, we just we looked at presumably duct vesicles, but not looking at anything obstructing or some kind of hole around the active zone.
Okay, then no further question. Then thank you again.